Good morning, everyone. Everyone here in the auditorium and everyone on the webcast online. This event is webcasted live. I'm really excited to open again for the 6th time the World Cancer Day event, which is hosted by the Cancer Biology Research Center at Tel Aviv University. I'll give you some grim facts together with some optimistic advances that occurred in the last year regarding cancer research, both whether it is basic or clinically and translational science. If we look at the projections for 2026, the projection for new cases in the US is 2.1 million new cases in the US, 626,000 cancer death in the US, and if we look at Europe, they are predicted to have 1.2 million cancer deaths this year. Together with that, we have some a very significant amount of deaths that were averted. If we start looking from 1991 to 2023, about 4.8 million deaths were prevented due to the advances in new therapeutic and diagnostic means. The leading sites, however, remain the same. It's still leading the breast cancer with more than 30%, prostate cancer in men with 30%, lung 10 to 12%, and colorectal cancer about 8%. The alarming trends are that there is a rise in young adults. We can discuss later what are the reasons for that, but there is a clear rise in new cases, about 12% new cases that are found in people under the age of 50. We see early onset of colorectal cancer, and in pancreatic cancer, we see that the survival has risen to 13%, but this is driven by less aggressive tumors. We know that there are several types of pancreatic cancer, but for PDAC, pancreatic ductal adenocarcinoma, the survival is still only 8%. Together with that, some optimism. There is a milestone in survival. We're celebrating a milestone of 70% for a 5-year relative survival rate. This is a huge increase in survival. If we look at our Israeli contribution of Israeli researchers and clinicians, I won't go one by one, but you can see the breadth and the broadness of all the outstanding science and breakthroughs that occurred only this year coming out from Israeli labs, whether they are in Israeli institutions, universities, institutes, and hospitals. In a diverse aspects of cancer research, proteogenomics, in immunology, and some advances, technological advances. If we look at some breakthroughs that were approved by the FDA, we have novel approvals. Only in 2025, 16 novel oncology agents. This is representing approximately 35% of the total of 45 new drugs that were approved. So cancer has a big chunk in FDA approvals. We're expanding options for breast cancer with new therapies and or same therapies but for new indications. One of the comebacks were ADCs, antibody drug conjugates, that had a hype in the beginning of 2000s and went down, and now there is a huge rise in several ADCs that are in clinical trials and some approved. New tyrosine kinase inhibitors, big advances in KRAS targeting. This has been a mutation that is abundant in pancreatic cancer, lung cancer, and some others, but novel techniques and computational chemistry allowed for small molecules, new small molecules that show really promising data from clinical trials. We can see some selected approvals that will come in the upcoming year, whether it is oncolytic viruses, PROTACs, protein degraders, ribotacs that enter the RNA binding degraders, and selective estrogen receptor degraders, and some dual bispecific inhibitors, whether small molecules or antibodies. Some new technologies as well, mainly focusing on cancer vaccines, T-cell engagers, and to name a few. There are the number of oncology companies has risen, and we see many of the big pharma as well as the small biotechs that are coming in. If they were not there, they are coming into it. And just without further ado, I would like to ask Professor Ariel Porat, Tel Aviv University president, to come and welcome everyone. Thank you, Ariel.
Good morning to everyone. I'm happy to open this special conference marking World Cancer Day. Its central goal is to raise global awareness of the disease, actually not one disease. I think that Ronit told me that there are about 300, right, types of cancer, and to encourage its prevention, detection, and treatment. There is probably no disease that causes greater harm to humanity than cancer. Half of us have been, are, or will be ill with cancer. But as in other cases, and quite paradoxically, a shared risk unites humanity. In a divided world full of wars and struggles, it is easy to unite around the goal of eradicating diseases with cancer at the forefront. Tel Aviv University, through the Cancer Biology Research Center, is proud to host this important conference for the 6th time, as Ronit mentioned. The center is currently the leading and largest academic platform in Israel for cancer research in all its aspects. What makes our center unique? First, joining forces, concentrating the research conducted on campus under one roof. Second, the broad medical network, close cooperation with 18 affiliated medical centers. Third, research power, over 80 research groups working tirelessly to deepen the understanding of the disease and develop innovative methods for diagnosis and cure. And fourth, multidisciplinary synergy, a fascinating connection between researchers from different disciplines, leading international institutions, and the pharma industry. I would like to deeply thank Professor Ronit Satchi-Fainaro, head of the Cancer Biology Research Center and head of the Gray School of Medical Sciences, for leading the organizing committee and managing today's conference. I am proud that a researcher of Ronit's stature is among our researchers. I would also like to thank MERCK and TEVA for their generous and ongoing support that made this event possible. Thank you to all the participants who are with us here in the room, and to everyone watching us on the YouTube live broadcast across the country and the world. I wish us all a productive, inspiring conference full of new insights that will move us one step further toward a cancer-free future. Thank you. I apologize, I have to leave, but I would watch some of the presentations.
Thank you, Ariel, and thank you for your ongoing support. And speaking of support, I would like to thank MERCK and TEVA Pharmaceuticals for their really ongoing support. This is the 6th time that they are supporting this event. I would like to thank Tel Aviv University and the one and only Dr. Judith Ben-Porat, with our new recruit, Dr. Leeat Keren, and thank you, and Ayelet Hashdi, and Anne Krinsky, who helped organizing this day. So I'm really excited to open with the first speaker, Professor Asya Rolls from the Faculty of Life Sciences here at Tel Aviv University, who will share with us her ongoing research.
Okay, so hello everyone, and first thank you, Ronit, both for this day and for being a force of nature in this field. It's amazing to watch. So I would like to tell you today a little bit about the way we are approaching this whole connection between the brain and the body in the context of cancer. And I think it's not very trivial to look at that as we now understand, of course, that cancer is a systemic disease. And we understand that microenvironment is part of the organism environment. But to start and break down the connections that happen in the systemic level, it's something that is still a huge challenge in every aspect of physiology, and of course in the context of cancer. And I will just try to give you some vignettes and the way we are approaching this problem. So first you can ask this, of course, every system, like the immune system, the tumor itself, every other component, eventually has to be synchronized by the brain. The brain, for the organism to function together. And one of the questions is, okay, so how does cancer is recorded in the brain, or is it being recorded? How is it affecting the brain? And in fact, in recent years, there are many more studies establishing connection between different mental health problems and cancer. And you can say, okay, but this makes sense, right? A person who just got diagnosed with cancer is going to have depression, is going to have many other mental health issues, stress. But the interesting thing is actually often enough, the depression, for example, comes even before the diagnosis of cancer. And this is just one example. It's very evident in pancreatic cancer with apparently people who, in many cases, the diagnosis of pancreatic cancer actually happens by psychiatrists who recognize that there is something weird about the depression of the patient. And then they start to refer the patients on. So this is something that we don't really understand how, and this is an active field of research in our lab. But also the other way around. One of the key features of cancer, especially the later stages of cancer, is cachexia. And in this case, which has many symptoms like loss of appetite, fatigue, altered energy expenditure, difference, of course, systemic inflammation, and autonomic dysfunction. And you say, well, but all of this can also be regulated by the brain. And indeed, for example, it was shown by a work done by Violet Ayres, showing that the vagal nerve, which actually is regulated by the brain, and the connection to the liver can alter the progression of cancer. There was another very interesting work showing that actually some of the symptoms of cachexia, this anhedonia, the dislike and displeasure of life and lack of motivation, is actually regulated by changes in the brain that occur due to changes in IL-6 levels that happen during cancer, and then changes in the reward system activity. So, okay, can it also, these changes that happen in the brain, can they also affect anti-tumor immunity? And this was a very active field of research in our lab, where we were focused on the reward system, the same reward system that is critical for our motivations, for our hedonia, basically. And it is regulating all motivated behaviors. And a key component of the reward system is the ventral tegmental area. It's an area that is very ancient, it's very basic, and it secretes dopamine throughout the brain. What was also known is that patients who are experiencing placebo, for example, or they have this positive expectation, have higher activity in the reward system. And our basic questions were trying to dissect whether this reward system activity can somehow affect immunity. And especially, and we asked it in many different contexts, but the way we can do it, we can now establish causality. How can we establish causality? Is by activating the reward system directly, and we are doing this with DREADS, chemogenetics. It's basically, we are implanting a receptor in the specific neurons that is now going to respond to an agent that we are going to inject. So basically, I have a remote control to activate these neurons and establish causally what is happening to the immune system. And we showed it in the past in the context of antibacterial immunity. We also showed it in the context of heart attacks and how it regulates, that it can improve recovery from heart attack. And also in a work that was initiated by Dr. Fahed Hakim and was led by Tamar Ben-Sha'anan and Maya Schiller when they were back in the lab, we showed that actually if we activate the reward system in mice that have tumors, we can improve the tumor outcome. And the idea was the following. We just injected mice with tumor cells, Lewis lung carcinoma or B16 melanoma. We had two groups of mice. In one, we activated the reward system. In the other, there were controls. And for 14 days, throughout the growth of the tumor, we could then activate the reward system once a day and see what happens to the tumor. And this was published long ago, so I'm going to be very brief about it. But basically, we could see that the tumors were smaller. And they were smaller due to several factors. But just to highlight, the reward system, we are activating the dopaminergic neurons in the brain. Dopamine doesn't cross the blood-brain barrier. This is why we use L-Dopa, right? So how does it affect the size of the tumor, which is in the periphery? So what we found is that it was mediated actually by the sympathetic nervous system. So we have the sympathetic and parasympathetic, which are regulated by the brain. And the effects that we were seeing were mainly regulated by the sympathetic nervous system. And more specifically, changes in sympathetic innervation to the bone marrow. And you can see here, this is just a staining for tyrosine hydroxylase, which is a key enzyme in the production of neuroadrenaline. And you can see that it is reduced once we activate the reward system. And who is listening to the signals from the sympathetic nervous system in the bone marrow? So what we found that one of the cells that has the highest levels of receptors to listen to neuroadrenaline are actually myeloid-derived suppressor cells. And interestingly enough, once they leave the bone marrow, they also remove these receptors. And of course, myeloid-derived suppressor cells are not the cells doing the effect. So who was the effector cell? We found that at least one of the effector components was CD8. Here you can see it in the level of granzyme B, that when we ablate the MDSCs, we now lose this effect. And these MDSCs were indeed necessary to mediate this response because, again, if we lose the effect, we now lose the beneficial effect of VT activation. So just to sum up, what we are saying is that reward system activation by changes in sympathetic activity, specifically reduction in sympathetic activity, alters the neuroadrenaline levels in the bone marrow and in turn affects the activity of myeloid-derived suppressor cells and results in changes in anti-tumor activity of CD8 cells. Okay, but this is all mice. And we have no idea if there is even any connection between what we are manipulating in mice, because as Tamar, who did all this original work, she used to say, well, we are just hammering on the reward system. Of course, we are going to see something. So we wanted to see if there is even such connection in humans. And what we did, I want to show you just what our first step toward proving that there is some connection between reward system activity and immunity in humans as well. So this work was done together with Talma Hendler and her student Nitzan Lublianker and my student Tamar Cohen. And the rationale was the following. We wanted to be able to control reward system activity. Now, how do you know in humans if you're doing anything to the reward system activity? fMRI. So we used fMRI-guided neurofeedback, which is Talma is an expert in this field. Then we wanted to be able to follow the change in their immune response. How do you do that? So we decided to give them vaccines. then we could time lock and compare each individual to its own time, to its own state. And after the challenge, we can then assess exactly the antibody titers. So what is neurofeedback? Neurofeedback is basically a way that the subject is entering the fMRI. He's given many different strategies, and individuals pick a specific behavioral strategy. In this case, what we found that, and it was in line with what was known before, having positive expectations, not planning, but actually thinking about positive experiences, activates the reward system. And once they're able to activate the reward system, we're giving them a score. So, okay, this worked. And people actually get better in activating this network. So we had 85 subjects. 34, we were targeting the mesolimbic reward system. Now, when I'm saying mesolimbic, it's much bigger than the VTA that I showed you because it also includes the nucleus accumbens, which will become important later on. This was our prediction that this is how it works. And they were targeted this. We had another group of controls that were random controls. So they were just given different networks, each one was working on a specific network, a motor network, other networks. And another group was left untreated. So subject would come, and for 3 sessions, they will do neurofeedback. They will train to improve their neurofeedback. It becomes overall 50 trials of neurofeedback, and people get better. We collect their blood in the last day when they come. We collect their blood, and then they are going through a neurofeedback session, and then we are giving them vaccine. And then after 14 days, we take again the blood and see what happens to the antibody titers. Of course, we can now do with in-subject comparison. And just to emphasize, neurofeedback itself is rewarding. So just the fact that people are expecting to get a score on of how well they activated their brain, it's going to be rewarding. So what we ended up doing is actually comparing the level of antibody titers and different components of the reward system, either VT activity or nucleus accumbens activity, which is another component of the reward system, versus the antibody titer. And what you can see is that we could see a significant correlation in the antibody titer to the activity in the VTA. Just to, again, these are very variable components, so just activation of the brain is very different between individuals, same antibodies, and still we could see this correlation with the VTA activity, which was very interesting because this is what we saw in mice, but also later on what we discovered that our original hypothesis that it's going to be VTA nucleus accumbens was also wrong in mice. And we actually probably had to do, I think if we're going to do it again, we'll do even much more focused neurofeedback on the VTA itself, and we can even get a better result. What does it mean? It doesn't move here the time, so I don't know if I'm okay on time? Oh, okay, good. So, okay, so what does it mean, just very briefly, in terms of potential for therapy, because I think I'm a very different bird in this crowd. First is that we now can target the organism in a very different way to try to modulate immune system activity in different strategies. And only this June, the FDA approved the first time vagal stimulator as a way to treat autoimmune conditions. And one of the things that we are seeing that also in our manipulation, we are affecting these peripheral systems, sympathetic and parasympathetic, also in humans. Other strategies that now emerge is actually modulating different brain areas by implants. And people have implants in their reward system for OCD. So it's not completely far-fetched, and we now have work with Ben Shofty in Utah in which we are manipulating, we are looking at different individuals that have implants in different brain structures, and we can collect their blood. And there are many other technologies that emerge on a daily basis in this field, focused ultrasound, magnetic stimulators, different non-invasive stimulators, and I think this is potentially going to be a game changer to the field once we start to look on the overall response as a synchronized one. So with that, I would like to thank the people who were involved in this work and people who contributed to that, and also say happy birthday to Yuda, who is working on the cancer in our lab and has a birthday today. So thank you very much.
Thanks, Asya. This was great. Questions? Yes.
I don't need a microphone. Sorry.
No, for you, but we need it for the recording.
It's for the recording. Okay, so don't think it's working. Okay, Asya, very nice. What about mood-modifying drugs? How do they affect, inflammation, cancer progression, and so on?
It's a whole very active field. Here, Shamgar Ben Eliyahu, for example, is giving noradrenaline blockers for postoperative responses. So, of course, the connection between just noradrenaline itself and immune activity has been well established. And I think these are strategies that are going to be very much applicable in the field. I think SSRIs on their own were shown in some cases to be helpful for the anti-tumor immune response, but the mechanism is unclear. There are some different strategies in how this can happen. And of course, there is a big question of whether this mood, whether what you need to change is something at the level of the brain, something at the level of the periphery. What are the implications of these moods? Are they an outcome of the cancer, or are they somehow also pushing the cancer forward? And some argue that there is even connection between brain activity and the relapses, the reoccurrence of the cancer. So I think this is all it's a huge potential field of starting to utilize drugs that were designed to cope with neuronal conditions and apply them to the field of cancer. Yeah.
Any other question?
Hi, beautiful work.
Thank you.
What about cognitive treatments or all sort of non-drug kind of treatment?
Yeah. Well, of course, once we started to move to neurofeedback is a non-invasive, and we did it very specifically so we could see, but of course, all the VR and all of this new world that is emerging is going to be very much relevant. And there is another component that I didn't mention here, but there's also the question of interoception. So how the brain represents the state of the body. And this is something that can be modified by different therapies, behavioral therapies, representation of psychological intervention. So I think there's a whole field there. The question, of course, are what the mechanisms. How eventually this psychological intervention translates into a change in brain activity, which in turn can then change something in the physiology. And this is the gap.
Great talk, Asya. I have just two small questions. Sports, I know Karmit is working on that, but does that also connect to the reward system? And
not in my case, but in others.
Yeah. And the location of the tumor, does it matter for the reward system or the type of cancer?
I think in general, well, Roy is going to hopefully answer that very soon, but the type definitely matters, because it's not only, I think what the brain is going to record is the metabolic demands, the effects, because the change in the immune system, all the other components, and this is eventually going to be recorded by the brain. And by the way, I think it's also going to be a way to detect cancers if you start monitoring the brain in the right way. So yes, I think there is we can get to a level of specificity, and the reason I think it's going to be relevant, because not all cancers have prodrome of these mental changes. Some are more and others are less. For example, breast cancer has more, and other femoral cancers have less. So it really matters. It's not just the type, not only the location, but also the overall, how it's going to be impacted. And we see that in many different indicators.
Wonderful talk.
Thanks.
So in the case of cancer, if at the end point, what really affects the cancer are the CD8 cells via this specific axis, why not directly affecting or perturbing the CD8 cells?
I think one of the things that we are discovering is new ways in which you can then target the downstream effectors. It's not necessarily that you have to go to the brain, but the brain can reveal new ways in which you can approach it. And for example, the changes that we saw in the MDSCs were not like any other that we could compare it to any other treatments. Something was happening in the way by neuroadrenaline, the mechanism, how MDSCs respond to neuroadrenaline somehow affected the downstream outcomes in a different way, and then CD8. So yeah, I think that once we start to perturb the system in different ways through the brain, we're just pushing it differently, and then we can also watch and see what the downstream effectors and go straight there. Absolutely. But also I want to tell you, we were looking for it, right? We were actually trying to explain. So I'm not sure if it's only the CD8. I'm pretty sure it's not.
Just one last question. With the hype of vaccination, due to the cancer vaccines were always a huge effort, but less successful one. And in a way, thanks to COVID pandemic, we saw some vaccinations being successful, and now we see a big comeback for this field. How would you design the vaccination today if you had to combine it with this approach?
Wow. I don't know if it's going to be the same strategies that you will take for every other. From here, for example, for us, vaccine was just a way to manipulate to see the change in the immune system. But of course, it's applicable. And then maybe you can boost the efficiency. One of the things that we are starting to look is whether the brain can somehow modulate the transition between innate and adaptive, the dialogue between these two components. So if there is a way to add, and maybe now some kind of beta blocker together with the vaccine can somehow improve the activity. I have no idea, but I think we can start thinking of that as part of our
resensitizing cold tumors into hot.
Well, thank you.
Thank you. And we'll move to the next speaker, Professor Tali Ilovitsh from the Faculty of Engineering here at Tel Aviv University. Thank you.
Okay. Good morning, everyone. I'm very excited to be here today. And first of all, I would like to thank the organizers for inviting me. And today I'm going to talk about the research that we're doing in my lab in the field of focused ultrasound and gas bubbles in order to treat cancer. I will start by talking about brain cancer, because in our brain we have a unique mechanism, what is called the blood-brain barrier, that aims to protect the brain from the delivery of undesired substances from the blood and into the brain. And the presence of the BBB is good when the brain is healthy. But when the brain has a condition such as a brain tumor, the fact that we have the BBB prevents the delivery of different therapeutic drugs into the brain. And what we want to do is we want to have a way to open the BBB in a transient manner only at the region of the tumor to allow the delivery of the targeted therapeutics there, and then after a certain period of time, the barrier will close. And in order to do that, we use focused ultrasound. So ultrasound in general are sound waves that propagate through our body. Ultrasound is non-invasive, It is safe, it does not contain ionizing radiation, and in diagnostic, it's the most widely used medical imaging modality. Now, ultrasound under specific frequencies, low frequencies, can actually penetrate through an intact skull. So we just take an ultrasound transducer, we place it in close proximity to the skull, and the sound waves then propagate through the skull, and we can create a focused beam. A focused beam similarly to a magnifying glass that can focus light into a specific point. We can do the same thing with sound, and then we achieve a focused beam at the right location. So how we can use focused ultrasound in order to open the BBB? To do it, we combine ultrasound with microbubbles. Microbubbles are gas spheres. Their core is composed of a gas, a heavy gas, it's a perfluorocarbon gas, and the shell is usually lipids. These microbubbles are already FDA approved as contrast agents in ultrasound imaging for over 30 years. 10 minutes after injecting these microbubbles, they are cleared from circulation. The gas is cleared through the lungs, and the lipids are cleared through the liver. And during the time that they're present in the blood, they are used as contrast agents. But here we're not talking about imaging, we're talking about treatment. Gas bubbles can also be used for treatment because when we apply ultrasound to these bubbles, as ultrasound is a pressure wave, when we change the pressure that we apply to gas, the bubbles will either expand or contract. And this is what you see here in this video of the microscopy image of a bubble. So we get these repeated oscillations under a specific ultrasound pressure that we apply. When we further increase the pressure, the behavior becomes more violent. The bubbles start to collapse and basically explode. And the thing is that it was shown that when we apply focused ultrasound to bubbles, when they're present in blood vessels inside the brain, the BBB opens safely and transiently, and then the BBB stays open for a certain period of time. And after that time, the tight junctions close again, and then the BBB restores its integrity. And there's an ongoing trade-off between the size of the BBB opening and the amount of pressure that we need to apply. When we increase the pressure, the bubbles oscillate more, and so the gaps in the tight junctions become larger, and we can deliver larger therapeutic agents. And if we apply low pressure, where the bubble oscillates in stable cavitation, it only oscillates repeatedly, we can deliver small agents. And the thing is that in my lab, we're able to find a unique set of parameters that triggers high amplitude oscillations of the bubbles to open nicely the tight junctions and facilitate delivery of large therapeutic agents across the BBB. So the first thing I'll show you today is a collaboration we did with the lab of Professor Dani Pe'er and also the lab of Professor Dinora Friedman-Morvinsky. And what we've done is to try and deliver lipid nanoparticles. It was in sponsorship also with TEVA, to deliver lipid nanoparticles across the BBB. So Dani's lab is an expert in lipid nanoparticle fabrication, and they had this paper where they used lipid nanoparticles and they stereotactically injected them into glioblastoma tumors, and they were able to prolong survival in this model. However, these lipid nanoparticles are 100 nanometer in diameter, so they do not cross the BBB, and they had to inject them into the tumor. And we thought that we have the technology to open the BBB, so how about trying to use a systemic delivery of lipid nanoparticles, and this is what we did. We tried to deliver different sizes, starting from 1 kilodalton all the way up to 100 nanometer mRNA lipid nanoparticles. And here you can see the ultrasound setup that we have. The mice are positioned on top of a water tank. The transducer is from the bottom facing up and targeting the brain. With the laser pointer, you see the region of the ultrasound focus. And after the treatment with ultrasound, we injected the therapeutic agent and then took the brains to microscopy, and we could clearly see the extravasation of the desired agent across the BBB. We targeted the right hemisphere here with the focused ultrasound and use the left hemisphere as the control. And we were able to see that we delivered both small agents and the large lipid nanoparticles, as you can see here. After doing this, we also moved and tried to do it in glioblastoma tumors. So we used a certain type of GBM cells called 005. These cells fluoresce in green. And you can see here via microscopy that focused ultrasound and lipid nanoparticles on their own didn't do anything. We didn't see any extravasation because focused ultrasound doesn't open the BBB. Only when we combine this with microbubbles, we get BBB opening, and we did it also. We were the first to show the delivery into glioblastoma tumors of these mRNA lipid nanoparticles. So this is in the field of brain therapy, but we can also use microbubbles and in general gas bubbles in the field of other solid tumor types. I won't go into depth into the introduction, but the idea that we had is that each tumor is different. Although the term is cancer, each tumor has its own biology, and if we wanted to fabricate a new drug, we will need a specific drug to each tumor type. But our method is a physical method, meaning that we take the bubbles, we excite them with ultrasound, and they trigger a bioeffect. So we thought that the treatment that we propose here could be more of a general way to treat cancer, similarly to surgery, for example. And here what we used is our different ultrasound parameters. For BBB opening, we want to maintain the bubbles only at stable cavitation, meaning that the bubbles only expand and contract repeatedly, because if we'll increase the pressure and the bubbles will explode, we can achieve microhemorrhage in the brain, which is undesired. But in other cancer types, what we thought is to use these bubbles as weapons, as detonators, that they will target the tumor cells, and then when we apply focused ultrasound, the bubbles will explode and they will fractionate the tumor. So this was what we did. However, there is a challenge with the microbubbles that I've shown you. Microbubbles are about 1.5 micron in diameter, meaning they're too big to extravasate from the blood vessels and into the tumor tissue, even if we include the EPR, the enhanced permeability and retention effect that causes the vasculature to be leakier. Microbubbles are just too big. So when we tried to do it with microbubbles, we had to intratumorally inject them. And the idea was to use something that is systemically injected. So for that application, we developed nanobubbles. Nanobubbles are just smaller bubbles. So if microbubbles are 1.5 micron, nanobubbles are 200 nanometer. So we were able to fabricate them. And then what we did was to systemically inject them into mice, tumor-bearing mice. In that case, it was breast cancer. The nanobubbles accumulate within the tumors. We applied focused ultrasound and then harvested the tumors, and we could see the generation of these lesions within the tumors that we treated them with nanobubbles. And as a control, we used microbubbles. And like I said, microbubbles do not extravasate, so we didn't see any effect with the microbubbles. Here, what you can see is that although we have these large lesions, some of the tumor still remains intact. And actually, we saw that this is actually an advantage, because when we have these tumors that now have these mechanical lesions formed within them, we saw that it enhances and amplifies the immune system to come and check what's going on there. So we wanted then to think about a way that triggers few complementary things in parallel. One is a method that would reduce tumor viability by just shrinking the tumor. Then we also want a method that will increase the immune cells within the tumor. And to achieve that, we thought about combination therapy and combining our nanobubble-based ultrasound approach with immunotherapy. And we tried different types of immunotherapies. The first one was anti-PD-1. And in anti-PD-1, we actually saw a nice recruitment of macrophages and CD8 T cells into both the directly treated tumor and also the distant tumor. So our model, we had 2 tumors, and we only treated 1. So this was with anti-PD-1. And the other combination that is currently in active collaboration is with Dr. Anat Globerzon-Levin from Ichilov, which is an expert in CAR T-cell therapy. And what we've done there is we applied focused ultrasound, and then a few days later injected systemically CAR T-cells. CAR T-cells are very well, are acting very good in hematological cancer, but not very well in solid tumors. And we were very surprised by the results that basically almost all the mice treated with a combination of ultrasound, CAR T, and nanobubbles, the tumors just disappeared. And this shows as a promising combination therapy, and like I said, it's an ongoing collaboration. So the last thing I'm going to show today is a new approach that we're currently working on, and this is a hybrid approach. If we think about the BBB and the research that I've shown you there with the lipid nanoparticles, the lipid nanoparticles, we had to do it in a 2-step treatment. First, we applied focused ultrasound and microbubbles to open the BBB, and then we had to inject the lipid nanoparticles. But how about generating a hybrid particle? This hybrid particle will be responsive to ultrasound on its own and also contain the therapy inside. So we moved to microfluidic nanoassembly fabrication, and we used here a different, not the core of these droplets is not gas. It's actually a perfluorocarbon of a higher order that under room temperature, it's liquid. So we now have microfluidic generation of ultrasound responsive droplets. Inside the core of these droplets, we can dissolve different therapeutics. These droplets are about 150 nanometer in diameter. And when we apply ultrasound, the perfluorocarbon core vaporizes and becomes a bubble. So these agents can become ultrasound responsive. And first of all, we fabricated these droplets. You can see here a TEM image of both a clean droplet and a drug-loaded droplet. You can also see ultrasound images of these droplets. So here we have never mind. Here you have imaging of only the droplets. The droplets are anechoic because their core is liquid, but when we apply ultrasound to the core becomes gas, and if it's gas, we can see it with ultrasound. So after applying ultrasound to it, we all of a sudden can see contrast that is generated. We also created droplets that contain fluorescent tracers. So when these droplets are in the droplet state, we do not see any signal under microscopy. But when we apply ultrasound, we can see that they become microbubbles. And then the last thing we did was also to look into the pharmacokinetics in the blood of these droplets. We were able to release drugs using ultrasound that is applied to these droplets. And lastly, we were successfully able to open the BBB with these droplets and release drug, all using a same agent. So I hope I've shown you some interesting concepts in the field of ultrasound, that we can deliver lipid nanoparticles across the BBB with focused ultrasound, that we can treat solid tumors by exploding nanobubbles within these tumors, and that combination with immunotherapy is beneficial in order to create a systemic response. So with that, I will conclude my talk. I would like to thank all the lab members and our funding sources and obviously our collaborators, and thank you for listening.
Thank you, Tali. Thank you. It was fascinating, especially to see the first 2 talks already so multidisciplinary, combining psychology, immunology, biology, and engineering now. Any questions? Yes.
So very nice. Inspired by the study you did with Danny Pear, can you control the composition, for example, of an LNP so it will itself become responsive to focused ultrasound?
So this actually what inspired us in the last project that I've shown you with the droplets. Lipid nanoparticles on their own, the core is not ultrasound responsive. You need to have something that acts differently under ultrasound application, and most liquids do not respond well to ultrasound because the acoustical impedance between liquid to the surrounding medium is very similar. So it's an engineering and chemical challenge to find the exact components, but with higher order perfluorocarbons that are liquid under room temperature, you can achieve that, but then you need to make sure and identify which molecules can be dissolved in these perfluorocarbon cores.
Yeah, so actually that's what I meant. So to affect the composition of the LNP, so in addition to the RNA that is encapsulated, co-package the gas that can respond to the ultrasound.
Yeah, so this is exactly what we are trying to do here. Currently, we encapsulated chemotherapy, immunotherapies inside, and ideally, we also want to try RNA, for example, but we want to see the stability of the RNA in such an encapsulation. It's also possible to do, for example, double emulsion and other techniques, so it's feasible.
Thank you. So you can focus the ultrasound to a specific site?
Yes.
And do you direct it to the, for example, in the brain, you direct it specifically to the tumor site or to more like choroid plexus sites or specific areas that are more permeable to begin with?
So the question was where we target the focused ultrasound. We can control the size of the focus. It depends on parameters like the frequency, the size of the transducer, and also the location. Here in cancer therapy, obviously, we're trying to focus it to the tumor region, but it can be anywhere, basically, in the brain. So we can do it. We are not limited by where it reaches the whole brain.
Hi, thank you very much for your thought. So I have a question. Like you show us that the main limitation for the lipid nanoparticle to reach the brain was their size of 200 nanometers. But if we look at the glioblastoma where the BBB is partially disrupted, and if we use like smaller particle, like polymeric nanoparticle that can be up to 100 nanometer or even less, do you think the ultrasound will have the same effect in enhancement of permeability and the BBB crossing? Like, did you check different size of particles?
Yes. So in general, the GBM is leakier than normal brain tissue. However, this leakiness is dependent on the tumor stage and also the location. It's not that the entire tumor is leaky. For us, in each tumor, we also injected only particles as control, and I think I've also shown it that, for example, the lipid nanoparticles didn't cross or barely crossed into the GBM tumor. So the leakiness is very heterogenic, and also it's not like very therapeutic dose effective from what we've shown or saw.
We just last question about translatability. How do you envision this as a treatment for humans?
So first of all, for BBB opening clinical trials, there are currently about 20 ongoing trials. So it is feasible, and it's going the way there. And we're
as immunotherapy rather than opening.
As immunotherapy, so it would require, passing all the stages of regulation. But if we are looking into, for example, an immunotherapy that is already approved, like checkpoint inhibition, and trying to combine it together, then that would be an easier path than inventing a new, obviously. So.
Thank you. Now we'll move to the next speaker, Professor Ofir Cohen from Ben Gurion University. See you. We will speak about deciphering cellular states underpinning metastasis and therapy resistance in advanced breast cancer. Thank you.
Thank you. So thank you, Ronit, for organizing this incredible meeting today. It's a great honor, and thank you all for coming. And so we are a very new lab, started in 2023, and what I'm going to present today is this is the agenda for today, a roadmap, but it's also kind of the arc or the journey of the aspects that we were focusing in our lab, starting from my work in my postdoc and ongoing. So kind of the first aspect would be to look at driver mutations and clonal expansion, moving to non-mutational mechanisms, cellular plasticity, then augmenting the view by looking at the tumor microenvironment and the immune environment. First, a vignette about immune suppression, and then about treatment for immune activation. Finally, I'll highlight the importance of sepsis as an entity of research in oncology, bacterial sepsis, and just end by a kind of a technological, computational breakthrough using H&E that can maybe augment or perhaps even replace next-generation sequencing. So journey starts with looking at mutations and clonal expansion. We think of two steps. The first step is the malignancy transformation from normal cells to tumors, and the second, metastasis and evolved drug resistance. And the point I would like to make is that we have a relatively detailed landscapes of drivers mutations in the first phase of the malignant transformations through efforts, heroic efforts like TCGA, but we don't have an equivalent effort for the advanced malignancies, for metastatic and drug resistant. And as you know, these are the later stages that are contributing much of what we think of as morbidity and mortality. We don't have those numbers, we don't have those budgets as a community. And so one of the main efforts in our lab was to harmonize and augment multiple metastatic breast cancer cohorts, including some unpublished data from Dana-Farber. And for each one of those metastatic breast cancer tumors, we have both whole exome and transcriptome. Specifically, Maya, in her effort to understand the landscape of driver mutations, she devised a new ensemble model where multiple different tools that can infer significant driver genes were used together in order to get a more detailed and a more robust picture of which are the driver genes that we should care about in metastatic breast cancer. What I would like to just highlight briefly is that there is high congruence or correlation between genes that are important in the primary setting and genes that are important in the metastatic setting. However, when we look at genes that are more important in metastatic breast cancer, it's not perfect correlation. What we see is many genes that are known to be important in cancer. That is potentially giving us an insight that in those advanced settings, even if it started as a breast cancer, the tumors can tap into additional mechanisms of pathogenicity. This is basically a broader repertoire known in cancer in other malignancies. Now, so previously, the power came mostly by numbers, getting a multi-cohort, approximately 800 patients. We can also get more statistical power if, in addition to the metastatic sample, we are able to obtain an earlier matched primary. And then we can use an evolutionary analysis and pinpoint on the evolutionary branch that moves from primary to the metastatic and drug resistance, the events that are most likely or more likely to be contributing to the clonal expansion and the fitness benefit in those drug resistant and metastatic settings. What Maya did here basically is took only metastatic breast cancer patients which matched primary, and then only kept events that are evolutionarily acquired, meaning that they're either not seen in the primary or seen with a much smaller clonal fraction showing expansion. And with this analysis, it highlighted key genes like RB1, CDKN2A, and RBB2 that are more important in the metastatic and drug-resistant setting. Okay, so the second phase in the journey is talking about cellular plasticity. Cellular plasticity is known to be important in cancer, but only in the 2022 update, it became one of the hallmarks in cancer. And so in an early analysis, when we looked at acquired mutations, we found two key modes: reactivation of the estrogen receptor and a bypass with receptor tyrosine kinases and growth factor receptors. Odaya, in her project, she was aiming to understand how these two key modalities of drug resistance are mapping into the transcriptome. So this is basically a transcriptional landscape of MBC where we can see two key archetypes, if you will. One activating GFR and the other activating ER. The important thing is that we can look at those signatures and predict progression-free survival, which can help patient management. So what we see here is that we have higher expression of the growth factor receptors. It is lower growth factor receptor. And just to compare back to mutations, if we try to stratify patients based on mutations, we still have a signal, but it's much weaker, showing that perhaps the transcriptome is closer to the phenotype and more powerful in distilling patient management. And now Odaya is broadening it basically to find an atlas of the transcriptional footprints that can become evident with any potential driver gene. Okay, so continuing with the journey. Now, in augmenting to look at the immune microenvironment, and this vignette is about immune suppression. What we are using here is single-cell resolution RNA-seq in metastatic breast cancer. Since it's metastatic breast cancer, we have multiple different metastatic sites. And the question is, what are the differences that we can look at the tumor microenvironment when breast cancer metastasizes to different organs? What I'm highlighting here is one specific modulation that we found in the liver. If we compare the liver metastatic niche to other sites, what we see is strong immune modulation and suppression. This is coming both to B cells, but focusing on T cells. What we see here is that T cells in the liver are much lower in cytotoxicity and antigen processing pathways. And this was shown also by others that tumors with liver mets have poor prognosis in response to immunotherapy. So that's suppression. Now, given the reality that our immune system can be suppressed, it is of great interest, of course, for the entire community to find options to reactivate the immune system. And this project that I'm going to discuss is with close collaboration with Professor Moshe Elkabetz, a mentor, a friend, and a colleague, and a joint PhD student of ours, Gregory Perlman. So to present the project, which again initiated with Moshe's effort, the attempt is to activate T cells, but using dual targeting. It's going to be a bispecific adapter, which we target both AXL and PD-1. And just to briefly present those actors, PD-1, I don't think it's needed a lot of introduction for the T cells. AXL is a receptor tyrosine kinase, but it's also very much related not only to a more advanced, more metastatic and mesenchymal state, but also to suppression of T cells. And it actually had been shown in multiple different cancers that targeting AXL can also improve response to immunotherapy sensitized. So that was the goal, getting a bispecific T cell engager that engaged both of them, the T cell PD-1 and the cancer cell AXL. And Moshe, and again, heroic effort of multiple years, were able to see that it is actually very potent, both in vitro and in vivo. What I'm going to focus on today are the bioinformatic analysis results that came later with Gregory. So this is again a joint PhD student, Gregory, and in the effort here, this is an in vivo model where there are three types of treatments that are given, either the bispecific antibody or a combination, giving both of them, or a control. And then spatial proteomics with collaboration with Leeat here can give us insights into both which cell types are present and their communities. So this is kind of an image that we can see from the Acquia phenocycler, and we have multiple markers here kind of delineated or delineating potential cell types. So again, the experimental setting, we have vehicle control combination, either targeting PD-1 or AXL, or targeting them together with the bispecific antibody. We have the representing image and segmented image. So after segmenting into single cells, we got approximately 2 million cells. We have 3 mice in triplicates for each one of those arms. And so the first analysis that Gregory has performed is to try and understand what is the impact of this treatment on cellular composition. And what he found is that there is strong elimination of cancer cells, and even more so for proliferating cancer cells based on Ki67. And there is augmentation of CD8 T cells, and even more so activated CD8 T cells based on granzyme. There are more results in this project, but in the interest of time and to allow time for questions, how much time I have? All right, yes. Okay, perfect. I'm going to move to talk about the communities in this project. And what Gregory had done here is to basically analyze single cells, but in the context of their immediate environment, calling those communities and then naming the communities based on the dominant cell types. And the question is, at the community level, what changes do we find given the treatment? So the finding here was that with the bispecific treatment, there is strong elevation of antigen-presenting cells, both in the context of tumor antigen-presenting cells and CD8 T cells and antigen-presenting cells. Okay, and so I'm going to skip now the results coming from human samples and then move to the last biological aspect that I wanted to emphasize. So sepsis based on infection is something that is, of course, of general importance, not only in oncology, but it is somewhat underexplored how sepsis can develop, or the manifestations of sepsis in cancer patients specifically. And so maybe I would say a couple of words about sepsis. This is a life-threatening, I would say, dysregulation of several physiological systems based on infection, usually bacterial infection, and it's life-threatening in general. But the percentage or the frequency of individuals that will develop that is much higher among cancer patients. So what we sought to do here is to devise a machine learning algorithm that can predict the development of sepsis specifically for cancer patients. So Dr. Ismail Masala is a PhD student in the lab, but he's actually already a board-certified oncologist. We started this research, and then he continued into a PhD, and it's a collaboration with both Leeat Multiple Medical Center and Professor Uri Obolsky from Tel Aviv University. What I would like to show is just 2 key results. Number one, which is, I think, kind of the highlight, is that we can predict that up to 48 hours in advance. And the reason it is important is that there are treatments that we can use, like vasopressors, and there is monitoring efforts that can be done. So in this case, if you do have early prediction of sepsis, it is very much has the potential to save lives. The emphasis here is that we don't use here sophisticated profiling. We kind of, I would say, the trade-off is instead of having very high quality in-depth profiles, we just use the normal clinical records, but we do that from multiple clinical centers in Israel, and we got upwards of 250 patients, oncological patients, and this is an analysis that was done based on the clinical records. the one result that I want to emphasize that when we look at the features within those clinical records that are most important, SHAP analysis or shapely value, we can see what physiological systems are being dysregulated. And more importantly, we wanted to see if we look at very short-term prediction, let's say 6 hours ahead of the sepsis, or long-term prediction, which gives you a very early indication. And we saw that there are some biological systems, like the kidney and the metabolic system and the immune dysregulation, that is coming as the canary in the coal mine, if you like, early signals. And we are excited. So this got kind of the first prize in the Leeat Innovation competition. And the reason we're excited about it is because we're trying to gear towards deploying that as a decision support tool in oncology to get kind of a head start on potential development of sepsis before the full clinical manifestation. Okay, so now in this journey, this is not a new aspect. This is just showing that, yes. Okay, so I'm just going to show that, that this is have the potential to perform multiple tasks, not based on next generation sequencing, but just based on histology. We can infer cellular composition from the work of Shalev. We can infer intrinsic subtypes of breast cancer. We can have very accurate prediction of the immunotherapeutic benefit score, and we can even get quantitative prediction for each single gene, which is much, much better than random, imperfect, of course. And just to say that we can glean insight into archetypes. If before it was done with a transcriptional map, we can build similar maps based on histomorphological properties. Okay, so quick summary from mutations to cellular plasticity, immune suppression and then activation, talking about sepsis in cancer patients, and then the potential of H&E. Acknowledgements, thank you all for listening. Collaborators and mentors, I mentioned Moshe and Leeat here. My group, and again, the people that done the work I mentioned throughout, and funding. Thank you so much.
Thank you.
Thank you
For this fantastic talk. Any questions? Yes, Yaara.
Yes, so thank you, very impressive. I have a conceptual question. So when you show the project, when you're showing this, you know, the Kaplan-Meier curves, and you say I can predict based on RNA, but it fails when I look at genetics, is that we have some patients that can be predicted based on genetics and others that failed? Or is it that overall, you know, in each patient, we can predict 70% of the response by genetics and then 30 is missing? So what exactly is the overlap between these two modalities, and what are we missing when we only look at genetics?
Okay, so it really depends on what is the genetic biomarker for. In this particular example, this is a cellular strategy, if you like, of activating MAP kinase through growth factor receptors. And then the way you would distill that for mutations is a little bit more tricky. It's not like you have one particular mutation that can give you this insight. I'm saying that because you can have basket trials that are really targeting one particular mutations and are very successful. What we think, and I think probably many in the field, is that if you look at the transcriptional footprint, it's basically giving you the steady state mirroring of all the changes that happen. It could be multiple mutations, it could be epigenetic, non-mutation, but you basically get a glimpse insight of what the cell is trying to do to overcome treatment and giving you a handle on what's going to be the kind of prognosis for that patient.
Just one last question. How do you compare the idea, mechanistically, of the bispecific T-cell engager of anti-PD-1 and the AXL compared to others that are T-cell engagers, bispecifics that are in clinical trials now?
Right. So this is a partial answer because I'm not sure if those that are in clinical trials have higher or low, but one benefit that was shown by Moshe is that it's not only targeting the PD-1 and the AXL, it's also improving substantially the interaction between cancer cells and T cells. It could be either...
So it's blocking antibody to PD-1
it's
or
blocking
non-blocking?
either, both, but additionally it is augmenting the interaction, making a higher interaction. And I suppose that bispecific antibodies, other bispecific antibodies can do that as well, but this particular molecule has strong signal towards this stronger interaction. Yeah.
Good luck.
Thank
you. Now, moving to the Weizmann Institute of Science, we have Professor Leeat Keren. We speak about spatial proteomics for next generation pathology. Thank you, Leeat.
עוד לא פרופסור.
You were made professor.
Promoted. I was promoted. Thank you for Tel Aviv University for promoting me. Okay. So hi everyone, my name is Leeat Keren and I'm from the Weizmann Institute. I'm very, very excited to be here. So far it's been a fantastic morning, Ronit. Really congratulations for the wonderful talks to all the speakers and also for having such a gender balanced conference. Okay, so our lab is generally interested in the tumor microenvironment. And the tumor microenvironment, as we've already heard a lot today, is very complex. It is comprised of many, many different cell types. And it's really these cellular interactions that then drive phenotypes that we and many in the community are interested in understanding. Things really like how do these multicellular interactions drive complex pathology. And so the method that we're trying to use to answer these questions in our lab is spatial proteomics that we've developed a couple of years ago, whereby we can take tissues and stain them with dozens of different proteins at the same time. And since this came out, again, a number of years ago, the field has really exploded with different methods, some of which we've contributed to. And last year, 2024, spatial proteomics was named Method of the Year by Nature Methods, and we were very excited about that. So basically, projects in our lab, I would say, are divided into these three main types. First of all, we have technological projects where we try and further develop these technologies to make them better, more accurate, higher resolution, be able to see more proteins. I'm not going to talk about this today. We also have projects on computation. So how do we take this very high-dimensional, complex data and turn it into biological insight? I'm also not going to talk about this today. And finally, we have projects where we take these technologies and actually apply them to study cancer. And even about this, I'm not going to talk about today, because what I thought to talk about today, and this is actually the first time that I'm presenting this, so very happy to get feedback, is on how we're now trying to really push this approach and take it from the research setting and into the clinic. And specifically, we're doing that in the context of lung cancer. So lung cancer, as you know, and it's been presented here, is a deadly disease. It is the second most diagnosed cancer, and it, of course, is the leading cause of cancer-related death. But the good thing about lung cancer is that in recent years, we've really seen an explosion of new treatment options for patients. So, if a couple of years ago we only had surgery, radiotherapy, and chemotherapy, today lung cancer patients have this ever-increasing and growing plethora of targeted therapies and many different modalities of immunotherapies. And this is, of course, great, but it puts a strain on the physician, which now has to answer a lot of questions when a patient presents, let's say, with a lump in the lung. Is this really cancer? Which type of cancer? And how should we be treating these patients? And to answer these questions, of course, this focuses on the pathology workflow, which starts from this, we try to do it as non-invasive as possible, so from a small needle biopsy. And then this material gets sent to pathology, where, again, the pathologist needs to answer a couple of questions. Is this tumor? For that, they'll do a diagnostic H&E. Then they need to say, is this lung cancer? Maybe it's a metastasis from a different organ. And if so, which type of lung cancer? And for that, they'll do a bunch of diagnostic IHCs. How should we be treating this tumor? And for that, they'll do both predictive IHCs and, of course, send whatever is remaining for next generation sequencing. And the problem with this workflow is that it is very it takes a lot of time. So the median time for diagnostic is usually a number of weeks. It's very wasteful in tissue material, and so about 15 to 20% of patients will actually exhaust their diagnostic biopsy and will require a second biopsy. This is an invasive procedure, and it also further delays treatment. And in this process, many of the patients, lung cancer patients, often present late, and so many of them will actually succumb to the disease before receiving a full diagnosis. And so from my introduction, probably you won't find it surprising that we would like to suggest a solution for that, which is to actually replace this very cumbersome process with a single multiplexed antibody panel that does all of these tasks simultaneously. And so to do that, we partnered with Hadassah Hospital, Raz Ben-Uri from my lab, together with Talkei Dareran and Eli Pikarsky from Hadassah, put together this 38-plex antibody panel that's geared towards diagnosing cancer and looking at targeted therapy, immune regulation, and others. We've put together a cohort of 106 cases. Out of these, 62 were resections, and we had 54 biopsies. And just look at these images of the biopsies here. I'm going to show you to them in multiplex. You can appreciate how difficult it really is. This is not easy material to work with, but the entire process was really optimized for these types of samples. So we took all of this cohort and we then diagnosed it using our multiplexed approach, where we stained for all of these 38 antibodies simultaneously, and we compared our results to the results. So here on the x-axis, you can see our multiplex diagnosis, and on the y-axis, you can see the original diagnosis done by the clinicians, and you can see excellent concordance between us and the original diagnosis. So we're getting basically the same diagnosis with less tissue. We had here two concordant, non-concordant cases. One, I think, really exemplifies what I said in the beginning. This is a case where we got the block of the biopsy, but there was actually no more tumor left in the block, which again exemplifies this issue of losing tumor throughout the process. And the second one was an interesting case where the original diagnosis said that this was a poorly differentiated cancer, meaning they couldn't assign a subtype, and we said that this was an adenocarcinoma. And looking at our stains, one of the things that helped us was that we saw this tumor was positive for ALK, which is a strong indicator of adenocarcinoma. And so we think that including additional markers can actually help us make a more accurate diagnosis. Okay. The second thing after we have the diagnosis is, of course, how should we be treating these patients? And one of the first things that's being done in the clinic today is, of course, to stain for PD-L1 to see if we can treat with anti-PD-L1 immunotherapy. And here again, this process is very challenging for the pathologist. Basically, what they need to do is they need to calculate what is called a TPS, which is a score that measures the proportion of tumor cells that are PD-L1 positive. And they need to distinguish between, let's say, tumor cells that are PD-L1 positive and macrophages that are PD-L1 positive, and this is non-trivial. So to do that, what they will do is they'll take one tissue section and do an H&E, and on the next one, they'll do the IHC for PD-L1 and basically mentally compare between the two to see where PD-L1 is expressed, okay? So this is very challenging because they need to mentally switch between slides. The task is even more difficult because the clinically relevant thresholds for PD-1 positivity are very low, right? So pathologists will look to define a sample as PD-L1 positive. You need either 1% or 5% of the cells to be positive, so you need a very high level of accuracy here. So again, we were thinking perhaps our multiplexed approach could actually facilitate this. So again, we took archival samples, we stained them using our multiplex panel, and this time we looked at the PD-L1 staining. We see this nice overlap between tumor markers and PD-L1 markers, so we can really quantify how much of the PD-L1 is expressed on tumor cells. And when we looked at these images, we said, okay, this is a pretty easy task now, so maybe we could actually automate it as well using all of the computational approaches that we develop in our lab. And so we did that. So we have now an automatic pipeline to understand who are the tumor cells, who are the non-tumor cells, and who are the PD-L1 positive tumor cells, and just quantify them directly. And when we do that, we can compare between our multiplex score, the automated one and the manual one, and you can see that we get 100% concordance. And if we now compare this to the original PD-L1 scoring, that was done by the pathologists. Again, you can see excellent concordance. We actually just had one sample that was different between us and the pathologist. And in that sample, the pathologist said that it was PD-L1 negative, whereas we said that it was PD-L1 positive. The reason was that this sample had 1.5% of tumor cells that were positive for PD-L1, and we could identify that. We then went to the clinical, so this is retrospective study. We went back to the clinical files of these patients, and we saw that this patient had squamous cell carcinoma of the lung. It was treated with chemoradiation, so they didn't give immunotherapy because of a negative PD-L1 score, but the tumor recurred. It was treated with nivolumab, which is, of course, anti-PD-1, and with good response. Okay, so we would like to suggest that perhaps we would have been able to suggest this treatment faster. And so I showed you this automated PD-L1 scoring. Okay, what about additional targets, right? So I showed you this very long list of targetable mutations and rearrangements in cancer. And some of these we can also identify using our approach. And I'm just going to focus now on two examples. The first one is ALK. So about 5% of lung cancer patients have ALK rearrangements. ALK is a transmembrane receptor. And what happens in cancer is that because of genomic rearrangements, it gets fused to a different components, basically stabilizing it and resulting in constitutive signaling. So in addition to this rearrangement, what happens is that there's also a huge increase in ALK expression in these tumors. And of course, we can identify that looking at the protein. And again, if we now take our multiplexed results and we compare them to results of ALK assessed by next generation sequencing, okay, because again, this is how it's currently being assessed in the clinic, we see that we get 100% concordance, and we can also identify this much quicker. Another test case that I want to highlight is the case of TROP2, which I think is very interesting. So TROP2 is a trophoblast cell surface antigen 1, again, a transmembrane receptor. And when we started this project a couple of years ago, TROP2, Ronit mentioned ADCs at the beginning, so there were just starting to come out ADCs against TROP2, and we were Googling and we kind of found that TROP2 was under clinical trials for non-small cell lung cancer. So Raz said, you know what, let's just, let's add it to the panel, see what happens. Fast forward 3 years later, 2025, these TROP2 ADCs got FDA approval. And last April, Roche got FDA approval for a companion diagnostic that is based on looking at the tissue. So one of the things about TROP2 is that when it's activated, it shifts from the membrane to the cytosol. So in order to understand whether it has a mutation, we can actually look at the ratio between the membrane expression and the cytosolic expression. And this is exactly the predictor, the biomarker that's been designated FDA approval by Roche. So they basically look at how much is in the cytoplasm and how much is in the membrane. Of course, they do this by immunohistochemistry, but we can do the same thing using our immunofluorescence. And here you can see the quantification in these two patients. Again, automatically we can say who are the cells that are TROP2 positive or TROP2 negative, and then try and apply it as a biomarker in the clinic. The last thing that I want to show you about this approach is that we think it's very beneficial and also facilitating integration of diagnosis and research. So in general, in lung cancer, but I would say even outside of lung cancer, the field is moving towards neoadjuvant treatments, which means that that people are starting to treat the cancer actually before the surgery. And this has many, many benefits for patients, which is of course most important. But one of the side effects of this is that we're actually starting to experience a shortage of non-treated tissue for research, because all of these patients are being treated with neoadjuvant therapy, and again, it will be beneficial for some, but not for all of them. So here again, we decided to add to our panel proteins and markers related to translational research, specifically to immune cells, because like I told you, this is what we're interested in our lab. We apply our full pipelines that we developed, starting from the raw data, cell segmentation, cell classification, to get these digitized cell maps. And from that, again, we can automatically generate this patient-level report. So this would be very similar to something that you get, let's say, from your blood tests. How many T cells, how many B cells, how many macrophages, only now you get this for each patient's tumor. directly from the diagnostic biopsy. You can see here, for example, the TIL fraction, which is, of course, important prognostic factor for immunotherapy. You can do cohort-level analysis, again, directly from the diagnostic biopsies. And here's just, one anecdotal result that we got from this cohort where we looked at neutrophils. This was because we're interested in neutrophils for a different project. And we saw here this increase in neutrophils in squamous cell carcinoma, for example, relative to adenocarcinoma. So just to summarize, I've shown you our suggestion for multiplexed imaging for one-slide diagnosis. We think that it has many advantages. It saves tissue. It provides more information for the pathologist to base their decision on. You can do automated scoring. I've shown you PD-L1, but this is true for other targets as well. You can easily integrate actionable and emerging targets. It enables looking at more complex biomarkers, so either things that need integration of several different proteins or looking at subcellular location, and it allows integration of diagnostics and research. And the big million-dollar question is, does this actually save time? And to look at this, what we're currently doing is we're actually setting this up as a prospective clinical study where we're trying to be rosh berosh against Hadassah pathologists and see who can deliver results quicker. So TBD. And so with that, I want to thank my amazing group for doing the work. This was led by Raz Ben-Uri and Tal Keder-Araran, close collaboration with Hadassah, but we have many other collaborators. Of course, our funding sources. And this is us in Purim, dressed up as antibodies targeted to, conjugated to heavy metal tags. So thank you.
Thank you, Leeat. Beautiful, beautiful talk. I have a question. I have many questions, but I'll just focus on one. The last slide that you showed, you put all the immune subsets on a range, as if they were like the blood count.
Yeah.
How do you define what is the normal range for it?
Yeah, so we don't have a normal range. Right, what we do is we take our cohort, so like among lung cancer patients, okay,
so it's relative.
Yes, but the more statistics you have, this is really what's relevant, right? Because the number of T cells in a normal lung is not as relevant as what you would expect in tumors, let's say for prognostication.
So when will it be out of range?
When it will be.
If the range is all the samples that you have.
No. So of course, no. Right. So you look at percentiles, etc.
Okay.
Yeah.
Questions?
Wonderful. Just a quick technical question. When you do this head-to-head comparison, you are actually using the same antibody, just metal conjugated?
So actually, we're not using here metal conjugated antibodies. We're using a fluorescence-based technology. I know, but it's such a good image. And this was the technology that we developed initially. But for this case, to be compatible with clinical-grade antibodies, we wanted to use the exact same antibodies that are being used in the clinic. So we're doing this with secondary fluorescence, and we're using a fluorescence-based platform.
Thank you for a beautiful talk. Two of the challenges of targeted therapies are the heterogeneity of the tumor on the one hand, and the selective pressure that targeted therapy exerts, rendering many patients resistant quite quickly. Does your approach cope with these problems?
Yeah. Oh, okay. So it's an excellent question. So for the first question, how do we do in terms of heterogeneity? So I'll say we don't cope with it any better or worse than the current pathology. The current pathology will take one section and diagnose based on that, and we do the same. We are preserving tissue, which means that you can now take this tissue and take it to additional assays. Let's say, if there's something that you're really worried about heterogeneity, you can take several slices, again, because you're preserving tissue. So I think this is one way to kind of start helping deal with this heterogeneity, but no, even with just taking this initial needle biopsy, you're already not sampling all of that, right? So this is kind of a given. The second thing is that patients quickly develop resistance to targeted therapies. And here, I think that our approach is less relevant. Probably, approaches that rely on liquid biopsies where you're monitoring in the blood and you're looking for mutations to reoccur, et cetera. I imagine these will be more important for this setting. Okay, thank you.
Thank you. Now moving to industry. I'm really happy to invite Professor Yanay Ofran from Biologic Design, the CEO of Biologic Design, who will talk to us about solving clinical problems with protein engineering, IL-2 as a test case. Thank you.
Thank you, Ronit, for having me here. And it's wonderful to see the great science that is happening here in Israel now. Let's see if I can find my slides here. It's open. Yanay, okay. Good. So I'm a computational biophysicist by training. Spent many years in academia, and I'm still affiliated with Bar Ilan University. But my move to academia, I actually moved to industry, I actually owe to ERC. I did not get an ERC grant I applied for, and then I decided I have to fund it in a different way. The reviews were excellent, but they said you can't do it with the $3 million you're asking for. They were right. So I went out of the academia just to pursue this project. And this project, back in 2010, suggested that we can use machine learning to design smarter therapies, to design antibodies that maintain the simplicity of the antibody modality, but they do things that antibodies that are discovered traditionally cannot do. And what I'm going to present to you today is a story. I will tell it in the scientific terms, not in the business terms, that implemented this approach on a very concrete problem. And this is an antibody that is now in phase 2 clinical trials, so I can also show you what happened in the clinic. So this is a story that many people who know oncology and immunology know very well. IL-2 is actually probably the first successful immunotherapy. Back in 1984, Steve Rosenberg at the NIH treated a 33-year-old patient with stage 4 melanoma with high-dose IL-2, and she is still alive today. She was stage 4. In 1984, it was unheard of that metastatic cancer can be cured. So this was a huge sensation, but you don't hear today of patients who are treated with IL-2, and the reason is very simple. IL-2 is actually a double-edged sword. It can bind to a receptor that is expressed on effector cells, expanding them, and if there is even one clone that identifies something on the tumor, it will expand that clone and will lead potentially to great efficacy. But it is also expressed, it also binds to a trimer that is expressed on Tregs, and if it binds to that receptor, then it expands Tregs, and Tregs will stop the activity of the effector cells. One Treg can stop between 10 and 100 effector cells. To complex things further, the affinity of IL-2 to the dimer is about 1 nanomolar. The affinity of IL-2 to the trimer on Tregs is about 10 picomolar. So any IL-2 that is in the system will first go to stepping on the brakes and only then to kill the tumor. And this is why IL-2, high-dose IL-2, is given in the ICU. Patients you electively expose the patients to a cytokine storm. Most patients have so bad side effects before they see any efficacy that you have to stop the treatment. So people since the 80s are looking for ways to solve this problem. The main approach that everybody is using is to engineer IL-2. If you can wisely and precisely engineer the cytokine itself such that it only binds the dimer on effector cells and not the trimer on Tregs, you solve the problem, right? So some companies went along with this approach, one of which was Nektar, promising results in phase 1, and then it was declared by the press as the greatest failure in history of clinical trials. Not long ago, a couple of years ago, a phase 3 that compared just anti-PD-1 to anti-PD-1 with the engineered IL-2 showed that just anti-PD-1 got better results. So the engineered IL-2 actually compromised the immunotherapy. And this was the most colossal failure, definitely not the only one. About 10 companies failed in clinical trials, and actually IL-2 now has quite bad rep because of that. Why? So immunologists will not be surprised by this. We showed it in several studies. What you do with the engineered IL-2 that can selectively bind to the dimer is expand the effector cells. And you know what they do other than to kill the tumor? They secrete IL-2. And they secrete non-modified IL-2 that goes straight to Tregs. So if you were exquisitely precise in your engineering of IL-2, you all but guarantee that you will create a negative feedback loop that will undermine your own drug. Supposedly no way out, right? So here comes the thinking about smart antibody. We thought, can we design an antibody that will grab endogenous IL-2? We don't want to give the patient exogenous IL-2 like Steve Rosenberg did. We don't want to give the patients engineered exogenous IL-2 like so many companies have tried. We want to give the patient an antibody, which on its own is inert. But we want this antibody to modify the IL-2 endogenously in a way that the IL-2 can only bind to the dimer on the receptor. If we can do this, we will expand effector cells. They will secrete IL-2, but we have excess antibody. We will grab this IL-2 and send it again to effector cells, blocking its access to Tregs. If we do this, instead of creating the negative feedback loop that undermines the success of everybody else, we will create a positive feedback loop, right? Easier said than done. This is the complex of IL-2 in yellow, bound to the trimer. Now, it has also been shown that the binding to the trimer has a different conformation than the binding to the dimer. The dimer is just the blue and the green. So the blue and the green are expressed on effector cells. The blue, the green, and the red are expressed on Tregs. What we need to do is, if we want to be successful, is to come up with an antibody that will not only block precisely the site of the red, CD25, but will also stabilize a conformation that is conducive to binding to the dimer. I will skip the AI part of how we designed it. I will only tell you that our approach is based on the following key steps. First of all, we computationally identify an antibody that does not do what we want to do, because if such antibody exists, then we have no job. But we predict that it's close to where we want to be. Then we use a set of tools to design a library around this template antibody. The library makes about 10 to the 8 or 10 to the 9 variants, each of them with 4 or 5 mutations, very carefully selected in predefined positions. We use this library not to search for the right solution, but to generate data to train a model. We train the model, and the model gives us the sequence of the antibody that does what we want, and then we can test it. One thing that this platforms allows us is to identify binders that bind one conformation but not another. So we can design antibodies that will ignore a pre-selected epitope if it's not in the right conformation, but will bind it if it is in the right conformation. Effectively, what it does is to tilt the ensemble of the molecule to the right conformation that we want, obviously. So, I now rotated the structure that you saw before. You still see IL-2 in yellow. You see the dimer, the green and the blue from both sides. And now you see CD25, the alpha subunit that is specific to the Tregs on top. I changed this view so that you can see where our antibody binds. So you see IL-2 in yellow here and in kind of turquoise over there. And you can see that our antibody sits exactly where the red subunit is, meaning it blocks the access of CD25 to IL-2. And it binds with very high affinity, single-digit picomolar. You remember that the trimer is 10 picomolar. So if there is a competition between the trimer and the antibody wins, even though it has a very small surface. Here you can see I'm rotating the IL-2 again. I took CD25, the red molecule, away, and I just marked the footprint of CD25. So the turquoise residues are where IL-2 binds to CD25. The green residues are where imnescibart, our antibody, binds. So you see it's very nice overlap, which is important. But what is more important, there is no hindrance to the binding of the dimer. What I'm not showing here, but if I had more time, I could show it. We are also stabilizing a conformation that increases affinity to the dimer. And right here, you can see in the SPR that if we bind IL-2 to our antibody and to the chip, and then we add CD25, which is the red subunit, there is no binding that you can see here. Here, CD25. When you add CD122, which is the beta subunit, it does bind IL-2. If we switch the order, we first bind CD25 and then first bind the beta subunit, CD122, and then CD25, there is no binding. So we spare the binding to the dimer, completely block the binding to the trimer. What happens in vivo? These are healthy mice, and you can see that with the dose response manner, our antibody expands CD8s, expand NKs, does not affect Tregs. So functionally, in healthy mice, we have the immune effect that we want to have. We also treated syngeneic models of cancer, of different cancers, and we saw good results. We also saw, not surprisingly, that it makes sense to combine this with other immunotherapy approaches, because what we do is to expand clones of specific recognition to the tumor. If you add an anti-PD-1, if this clone can be inhibited by the tumor, you overcome that. So that was encouraging. Another important fact that we learned during this process is that the antibodies that are designed by our platform have good developability. Now, this is not a word that is commonly used in academia. Itai knows it well, but people who don't study antibodies specifically are not aware of the fact that a molecule that behaves really nicely in the lab may not necessarily, and with all likelihood will not be a good drug, because the drug has to be producible at high titers. It has to be very stable. It has to be a molecule that could be taken in a box from a factory in China, put on a truck that drives for 4 hours, get on a boat, get to the other side of the ocean and still be stable. It has to be not immunogenic and have good half-life in humans. So a lot of requirements that have nothing to do with the huge task of binding in the right place and creating the right biological effect and having the right affinity. Above and beyond all that, it has to be a good drug. So we made this drug, and it behaves really well. It's very stable. It has good yield. So that was a good sign. By the way, for those of you who think about taking an idea to the actual world of treating patients, just some fact that people are not aware of. The cost of designing a molecule and testing it in vivo is about 2 to 3 million. Just making it at GMP to go to the clinic is about 10 million before you treated one patient. So these are the challenges that you have when you don't rely just on ERC. I didn't know it back then. Okay, what did we see in the clinic? First of all, these are all the drugs that we know of that published clinical results based on modified IL-2. And what we're looking at here is what happened to Tregs. All of them expanded Tregs. Imnescibart cut Tregs by half, which is not major inhibition. We don't want to get rid of Tregs. We just don't want to expand them. We treated some patients. Now I'm showing you just data for melanoma, late stage in phase 2. All these patients are refractory to checkpoint inhibitors. They did not respond to Ipi/Nivo or to Keytruda, and so forth. What you're seeing here in the waterfall plot is their response in terms of change in tumor diameter. So you see one patient had a 43% growth. Yeah, that's the good way to treat these results. All the others are what we call disease control, meaning either the tumor is stable or it's shrinking. And on the spider plot, you see how long this response lasted. So you see we have one patient that went quickly to 0. He's now already at 26 months, so 2 years with no detectable disease. And others that are being treated for a long time, these are patients that currently have no other treatment because they failed first line. Some of them already failed 6 and 7 lines. Just one example of one patient. You see here metastasis in the liver, quite difficult to treat. Circled in yellow, you see the shrinkage after 8 weeks. 2 of them already disappearing after 16 weeks. and after 24 weeks, just a little stub left from one of them. What happened to safety? So I can tell you that this is an extremely safe treatment. We didn't see any of the symptoms that are typical of a cytokine storm because we did not create a cytokine storm. So in that sense, just engineering, precise engineering of the molecule and the structure and the interaction can solve a clinical problem if we understand the biology around it. Thank you for listening.
Thank you, Yanay, for a fantastic talk and a fantastic journey starting from design up to clinical results. Itai, of course.
So this is fascinating, of course. 35 years ago, so antibodies for CD25 were developed. At the time, Tregs, I believe, were not known even. So it was believed that in certain hematological malignancies, CD25 was overexpressed, blah, blah, blah. Now, the best one, I think, was FDA approved. It was about 1 nanomolar, not picomolar. But now that we are appreciating the importance of blocking Tregs, so why don't we see, or why shouldn't we see a revival of neutralizing antibodies that will prevent binding to IL-2 on the Tregs?
That's a great question. So there was an approved anti-CD25 antibody that was actually retracted. It was removed from the market. And I think the reason is, and this is true for all cytokine-based therapies, if you block the cytokine, you know what signal you block. If you block a receptor, the receptors of cytokines tend to combine to bind different things. So CD25 is probably not dedicated only to IL-2, and if you block CD25, you may block other signaling. Above and beyond this, the antibody that was approved led to depletion of Tregs. And you don't want depletion of Tregs. You just don't want to increase Tregs. So this is why this approach of blocking CD25 did not work, and I don't think it will work.
Hi. So are you guys worried about immune unrelated adverse effects? Because we know that anti-PD-1 can result this type of adverse effects, and as you showed, it really increased the Tregs, and you are decreasing the peripheral Tregs.
So what we do is, first of all, are we worried? Of course we're worried. Always. But what we do is to starve Tregs. We're not depleting Tregs. So where there is an increase in IL-2, we will make sure that this IL-2 goes to effector cells. By the way, IL-2 is not the only mechanism that controls effector cells. We don't touch effector cells, and our efficacy is bound by the concentration of the antibody. So once the antibody is saturated, the IL-2 will go to Tregs, and the Tregs are still there. So in that sense, we think that our effect is limited to places where IL-2 is present and where Tregs play a key role in stopping the immune clearance of the tumor. So I think tumors where we know the Tregs play a key role in protecting the tumor, like melanoma, like ovarian cancer, like RCC, are tumors where we expect to see good results. We don't see widespread autoimmune effects because we don't give IL-2. We give a molecule that is inert and redirects endogenous IL-2. I did not show, but we have complete responses in head and neck. We have very deep responses in lung cancer. We have complete responses in bladder cancer. So other than GI and pancreatic cancer, in every tumor type that we treated, we saw these responses, and the safety profile is milder than checkpoint inhibitors.
Thank you. Great talk. I want to continue, actually, Itai's question. I know that CD25 is very important for T cell and B cell development, and I understand the safety issues in blocking completely CD25. But since you showed that you can actually direct the antibody to a particular recognition site, why not block the interaction between the CD25 and the dimer of IL-2 receptor?
So you're suggesting, again, treating from the cell side, not from the cytokine side. And this is an interesting idea. It will be more difficult to design such antibody because if you want to bind closer to the membrane, it will not be easy, and you will need to bind with a very high affinity, probably affecting also the beta and gamma. So I'm thinking out loud. We did not explore this possibility. It will also possibly lead to depletion of more cells because you will bind beta and gamma on all cells. So we didn't think about it, but it's an interesting idea.
Thank you. That was incredible. I have a question which relates to the part which you kind of glossed over. You mentioned that you had a library of about 10 to the 8 variants, and you trained the model based on that readout. So my question, based on your opinion, or maybe you tried, like the newer AlphaFold bolts and tools from David Baker, do you think that that could make a dent in that number, or there is no replacement to actually having millions of variants in order to train the model?
So in deep learning, there are basically two steps. There is what is generally referred to as pre-training, training a foundational model on vast, vast amounts of data. And there is what's called post-training, which is training, or something, it's called fine-tuning, which is taking a smaller set of data and gaining better predictions or better generation of relevant instances in a very specific domain. What we are seeing now is that we have kacha models for the general problems. Ronit is kicking me out, right?
Just if you want coffee break.
And I think that we will get to a point where we have a general model that gives us something reasonable, but the fine-tuning, I don't see in the next, I say, 5 years that we will be able to overcome this. We do use some foundational models, but the fine-tuning is key.
Thank you, Yanay. This was really incredible. I would like to thank all the speakers of the first session. We'll go for a break, and then we'll have the second session. I would like to ask all the speakers from both sessions to come for a picture here. And again, I would like to thank all the members of the CBRC committee that are here and at home, and to thank, now that Larissa and Dana are here, so we are so grateful to TEVA Pharmaceuticals and to MERCK for supporting this event for so many years from its beginning. Thank you. Really happy to invite Dr. Yaara Oren from Tel Aviv University, who will speak about rising from the ashes of treatment, characterizing and targeting persister cells. Thank you for coming.
So thank you, everyone that stayed, despite the fact that there is no free food available after this session. And a very sharp transition from what we heard so far. We heard some excellent talks of how things are working in the clinical world and how we're thinking of translating innovative technologies and computational methods into the clinic. I want to give you the sharp contrast of how things are working when it comes to basic biology. So what started the research I'm going to tell you today is not some major unmet clinical need. It was a simple phone call that I got 4 years ago. And on this call was a brilliant scientist named Charlie Falco, who I had the pleasure of training with. And Charlie, after finishing his PhD, transitioned into industry, and he was working at Bristol Myers Squibb, and I haven't spoken with him for more than a year, and suddenly I got a phone call, and he's like, Yaara, because that's how they call me in the US, because they don't have an Ein. And he said, Yaara, what do you know about CDK4/6? I was like, Charlie, I'm very happy to hear that you're healthy and well. Frankly, not much. And he said, you should be ashamed of yourself. I was like, okay, why? And he said, because CDK4/6 inhibitors are probably the most beautiful example of how basic biology translate into an amazing, very potent drug. So CDK4/6 inhibitors have been out there in the clinic first in 2015, and very quickly they advanced to be first-line therapy for ER-positive breast cancer. And the idea is that they translate very simple textbook biology to big effect on patient lives. And what these drugs do, they drive a very durable cell cycle arrest, which is exemplified by the fact that the cells enter senescence, which is a stable cell cycle arrest that happens to us as we age. And because they're so effective, very few years after they were approved, they were already moved to be first-line therapy for ER metastatic breast cancer that we heard so nicely from Ofir, how this is still a big, big thing that we're fighting to solve. Okay, so I said, Charlie, really thank you for this brief intro. That sounds amazing. So why are you interested? Because you now work in a pharma company. And he said, here's the thing. If you look now at the responses of patients, we have most patients respond very well initially to CDK4/6 inhibitors, but sadly, as was repeatedly mentioned here today, we see recurrence. So it seems like also in the clinic, these cells are entering senescence, but ultimately, we have patients relapsing. And when we look at these tumors that we sequence and we do profiling, because it's a pharma company and they have a lot of money, at about 30% of the patients, we do not see any genetic mutations associated with is very, very clear relapse. And I said, okay. And then he said, Yaara, doesn't that seem to you like this relapse is driven by persister cells? And now he caught my attention, because persister cells is in the heart of what my lab does. So what are persister cells? Persister cells are a rare population of drug-tolerant cells that can survive therapy through a non-mutational mechanism. And the hallmark of persister cells is, despite the fact that they are resistant to therapy, their progeny will not have any acquired genetic mutation, and themselves would remain sensitive to the same therapy. Okay, so Charlie was wondering if it can be that what they're seeing in the clinic is driven by persister cells, and this is why they fail to capture it using the mostly mutational profiling. So the truth is that when he first spoke with me about that, I said, you know, Charlie, I'm just moving my life back to Israel. I don't really know much about this field. And then he said the magic word that no scientist can ever resist. Yanay, he said, I'll fund you. And I said, so I'm in. So this work started while I was a postdoc, or was just leaving my postdoc. And it's a collaboration between my amazing postdoc mentor, Joan Brugge, that was a postdoc in her lab that started the work, Abram, and then when my lab opened in Israel, Itai took over this project, and he's really been leading both the experimental and the computational side of this work, together with other members of my lab, and Noah on the experimental side, and Marina on the computational side. Okay, so we had to start from scratch. So where do you start? You take a cell line model and you say, can we see the same dynamics that is seen in patients if we just add these inhibitors in vitro? So what we did is we took a ER-positive breast cancer cell line, and you labeled it with GFP, And we treated it with a combination of an ER blocker and a CDK4/6 inhibitor, because this is how patients are treated. And what you're going to see here is a video, and we're taking image of these cells. And you can see that throughout treatment, at the beginning, the cells are largely arrested, but after about a week and a half of continuous treatment, we do see that some cells are able to expand. So if we look at this time course, we can very easily define 3 different states. We have the naive cells, we have this kind of lag phase where cells become senescence, that in vitro, unlike in patients, it's very easy to detect because it has clear morphological changes, this kind of fried egg phenotype. And we also see that after about 2 weeks, we can see an expanding phase. And as you would expect, again, there are a lot of things we can easily do in vitro, but are much harder in patients. We can stain for beta-gal, which is the canonical marker of senescence, and we see that it's increasing with treatment. Okay, so we have this model. The drug induces senescence. Of course it does. It's a very effective drug. But is it reversible? Maybe. We just have cells, right? They enter senescence. I don't become any younger, unfortunately. Maybe you do. Once you enter senescence, it's a terminal state. So can our cells escape senescence, and does it seem to be reversible? So what we can do is we can do a trick that is commonly applied in persister studies. We can take cells that were persisters, that became these senescent-like cells. We can then remove the drug and see if they're able to recover, and take the population that recovered and re-expose it to the drug. Now, if whatever was driving this phenotype initially is reversible, the cells that are persister-derived should be as sensitive as the original population. But if they acquired a genetic mutation, then they should stop responding to the drug. So we did this. We derived persister-derived cells, and you can see that the persister-derived cells remain sensitive to the CDK4/6 inhibitor and also to the ER therapy we were giving. So whatever was driving this senescence is reversible and is not encoded in the progeny of these cells. Okay, so what drives it? So we could do many things because we had very generous pharma funding, but what we decided to do is to do single-cell sequencing. And the reason was that we thought that whatever is driving this effect is going to be a very rare population, because the vast majority of cells do enter stable senescence. And we thought that if we could look at the transcription over time at a single-cell level, perhaps we would be able to capture this very rare cell population. So what we did is we took our cell line and we treated it and we subjected it to single-cell RNA sequencing before drug administration at this lag phase, and at day 15 when we have these proliferating cells emerging. So this is how it looks like, and you can see each dot here is it's a UMAP, so each dot here is a cell. And not surprisingly, you can see that the transcriptome is shifted with time, right? We add a very strong drug pressure. And here are the treated cells, and here are our naive cells. And this is not something we were expecting, because why do we have 2 clusters? Okay, this is not patient data. It's a very nice, simple cell line. What's wrong here? And Itai has spent many sleepless nights about this cluster, and I also want to help Ofir for helping us troubleshoot this, that understanding that this is not a technical artifact. This is 2 differently genetic clusters. So when we work in a cell line that was derived from a patient, no one promises that it's going to be genetically homogeneous. So in our cell line, we have 2 distinct genetic subclones. Both of them are very different from a normal tumor. They have multiple copy number and gain events, but they're stable. Okay, so that's what we have. We were hoping to work on non-genetic resistance, but here we got also a genetic model, and this is what we have. Okay, so now we can continue and look at these cells and cluster them in a very, I would say, traditional way for people who are doing single cell. And we see more or less what we would expect, that before drug, the cells are showing proliferative markers. They're highly proliferative. It's a cancer cell. And when you look at later time points, you see these clusters that are associated with senescent. Only thing is that a part of these very obvious clusters, we also detected this cluster, which appears late, only in the untreated cells. And this cluster shows positivity for both markers. It has both signatures associated with being proliferative and signatures associated with being under drug, including senescence and interferon gamma response. How can that be? Is this hybrid cell state real? So we took advantage of the fact that we can have very good markers for senescence and for proliferation, and we double stained, and we saw that indeed in the proliferative state, cells are proliferating. At the senescent state, they're senescent. But under drug, we also have a very rare population of double positive cells. They're both senescent and proliferative. And the thing is that this population is super rare. Even in later time points, we can get only about 6% of them, but they're there. Okay, where did they come from? They should not exist. What we did is we used, again, a computational approach because we were lucky to have our system barcoded. So we can look at each cell and ask, are the cells from the same subclone, do they behave in a similar manner? And what you can see here, so each row, each column is a clone, you can see that when we look at a family of cells, we have cells that are clones that can give rise to these hybrid cells, but we have clones that are completely senescence committed. Okay, so it's stochastic, right? It's not. We can repeat this experiment again with this with the same parental population, but treated them separately, and you can see that this hybrid cells, the ability to give rise to hybrid cells, is highly predictive. So only some cells are able to give rise to these rare proliferating subclones. Okay, so now I know, because there are many people here studying genetics, that you say, oh, heritable genetics, right? And you know, there is always this fight between what matters more, genetics or epigenetics. And just to reconcile, both do. Okay, so we can take now, oh, there is a Mac to PC, but never mind. So believe me, on my Mac, it looks very nice. Okay, that both genetic backgrounds can give rise to these rare hybrid cells, but in different propensity. So conceptually, you can have very different genetic backgrounds. Both of them can lead to the same outcome, but the difference not if this outcome occurs or not, but in what fractions. And this is what we can see when we stratify this based on genetics. So, okay, great. So we know we have a trait. We can predict it is affected both genetically and non-genetically. But can we predict, right? Because at the end, although we do basic biology, we're interested in ultimately translating this. Can we predict which cells, which signatures will give rise to hybrids? So I want to say that usually we're trying to compare on drug to naive cells. But what we're trying to understand is, in the naive cells, what makes them different? What gives the ability, not of me to be resistant, but of my progeny in many generations? What is what is the inherent trait that enables some cells to give rise to cells that will ultimately be these hybrid cells? So we use a computational approach to map fate back to our naive population. And now we can do the simple gene expression, the simple differential expression between, done only at the naive cells, okay? Between cells that are senescence committed, lineages that are senescence committed, and lineages that give rise to hybrids. And now we have genes. This is the classical analysis we do, right? We have genes, but they don't reflect state. They reflect fate. And we can go and we can test them. And we can see that if we overexpress genes that prime for fate commitment, we can make our cells more committed to becoming stably non-proliferative and under drug. And just because I have one last 10 seconds, I want to say this is a very simple approach, but it translates very well. So this is just to show you that we're actually abolishing this double hybrid population. We can generalize it very quickly because now we have genes. So we can go and we can check them across multiple cell lines, like the CCLE, and see that indeed we can predict fate under drug. We can even take them now and look at patient data and stratify them according to initially what is the commitment score of the patient. And you can see that patients that have high commitment score have low risk of recurrence versus ones that their cells are not committed and they're more likely to recur earlier. So I want to end on a philosophical note. We usually compare resistant to naive, and we ask what is different. We do it for many years. We do it with genetics, we do it with RNA, we do it at single cell, but we're always asking the same question. We can ask a different question, which is arguably what we want to ask. What at the beginning makes one cell sensitive and the other resistant? Not what happens ultimately, but what predicts your fate. And by doing so, we might uncover completely new targets for therapy. And with this, I want to end. I want to thank Itai, who did the vast majority of the work, as well as the other team members, and thank you for listening.
Thank you, Yaara. This was beautiful. Questions? Any? Maybe I'll continue with your philosophical, and if we're thinking of the heterogeneity of tumors, and we know that some will become resistant and some no, and we want to select the ones that will become resistant and eliminate them. Do you think this is really viable in terms of, we do not have the prerogative of having single cell clones in a tumor, so how will we discriminate between them in a very early stage, right? Because once they are metastatic and resistant, it's less relevant.
Right, so I would say that there are some computational tools. I think you can think of the data set that Ofir has shown at the beginning, where you can try to do trajectory based on pre and on treatment and pre and post treatment, and then try to envision or to give a score to what you think would be the transcriptional trajectory. So of course we don't have single cell, we don't have this kind of lineage tracing resolution in patients, but I think some of these concepts of fate mapping and trajectory mapping can be done also in clinical data.
Thank you, Yaara. That was great. So you mentioned initially that CDK4/6 are approved for metastatic ER positive. I'm wondering to what degree you see overlap between the signature that you found to propensity for hybrid or commitment to senescence and ERness. So for example, T47D and MCF7, they're both ER positive, but with very different levels. So is it entangled? Is it like the commitment of the lineage to ER, or is it completely different?
So we didn't try to see if this works. So the clinical trial I showed you is specifically patients who are treated with CDK4/6, so they are all ER positive. We didn't try to stratify into, A, we didn't try predicting across other cancer types, and B, we didn't try to correlate this with ERness. But I don't think any of these markers, and we can ask Itai, is actually directly tied to ER, if that makes sense.
Yes, one last question, Adi.
Yaara, thank you for this excellent talk. Maybe I missed it, but did you show that those hybrid cells are more associated with resistance? Maybe I missed it. And I wonder if you can go to patients, a clear group of patients undergoing a therapy and being resistant, and try to check your hypothesis that they do have more hybrid cells, and that would strengthen. I think this can be easily done.
Yes, so I think there is, I think it would be amazing to do this, to see if we can see hybrid cells in patients. There is, for single cell data, okay, I'll start with the challenge. So for this, you need single cell data of patients who are currently on therapy. And this is not something that is publicly available, and also is not something that is clinically usually done, sampling these patients on therapy and then subjecting them to single cell profiling. You can ask Itai, we have looked, we have talked to labs around the world. Just the reason is that there is currently no clinical reason to do this, right? So we were talking about Leeat before, about what kind of samples we usually get. We don't have access to these samples. But I would agree with you that if we had, it would be very, very interesting to look at. What we currently have, the closest thing we have is bulk RNA, but then you cannot do this, that is on treatment. But then, of course, you won't be able to discern this rare population. So I would love to have it, but unfortunately, we haven't found the right people yet.
We can chat later.
That's why we have clinicians in the audience. Thank you, Yaara. So, thank you. Now moving to the Technion now. Professor Yosi Maruvka, who will speak about MLP-DT, a machine learning model for predicting doubling time from transcriptomic data. Thank you, Yosi.
Okay, so I'll just move on. Yeah. Okay. Sorry for the non-inviting titles, and thank for the organizer for inviting me. Great talk. So basically, I'm going to speak about tumor growth rates. And it is well known that growth rate is associated with cancer response, patient survival. Actually, in Hebrew, it works much better, so I'll just say one quick sentence in Hebrew. הדבר העיקרי שגידול עושה זה לגדול. Like in Hebrew, it's like the main thing that a tumor does is to grow. And so it's obviously a very important feature that we would like to know about our tumors. However, it is very challenging, or accurately almost impossible in almost all cases, to measure tumor growth. So hypothetically, what we would like to have is some kind of a serial, through time, ways to infer tumor size. And for that, we can infer the growth and can say if it's a very severe cancer, much more benign, etc. But we cannot do it. Why? Because almost always we start treating the patient right after we observe the tumor. So we're not trying to wait and to see, okay, what's going on, what will be your growth rate. No, we are right away trying to treat it and basically eliminating the ability to infer this very important information. So what is usually being done? We are trying to evaluate and approximate the tumor growth rate with some kind of proliferation biomarkers. The most commonly used one is Ki67, which is basically a protein that is active in most cell cycle phases, not in the same level, but in all of them, besides the G0 when cells are not dividing, so it's their naive time. However, proliferation and growth are very linked to each other, but are not the same, because in order to grow, a cell doesn't need just to divide very quickly. It requires also that most of its daughter cells will continue to live, grow, and divide. So proliferation biomarker is nice, but what we really want to have is some kind of a growth biomarker, which we currently don't have. Okay, however, there is a situation that we are able to measure the actual growth rate. It's not of the tumor, it's cell lines in the Petri dish, right? So we can do it, we can measure it, and then we can understand and know what is the exact growth rate, which again is a convolution of the proliferation and death. So cell lines in a petri dish has many advantages. It's very easy to measure, right? It's like one of the first experiments kids are doing in high school biology, just to measure the growth of the bacteria in the petri dish. We can replicate it, so we can be much more convinced in the fact that we measured a true measurement. And for us as a computational lab, it's very nice because we can already have this data was generated by many data sets along with a lot of molecular information. However, obviously, it has some little big disadvantage. It's a very different condition from what we have in the body, right? Like cell lines and tumor in the body, very distinct beasts. But we wanted to see, okay, let's see how far we can get from trying to train all our models based on cell lines in a petri dish, and after it to try to apply it to patients. So we developed a tool, we call it a DT learner, doubling time. I'm not going to get into all the technical details here in this talk today. It's not the audience for the technical details, but we found that the doubling time measurement that we had for the cell lines were more accurate than the growth rate, even though hypothetically it's just one divided by the other one. It was easier for the models to infer the doubling time instead of the exact growth rate. So basically, we start from analyzing cell lines, try to infer the features that are relevant. We tested it, we fine-tuned the model, we applied it to PDXs as a validation, and then applied it to patients. Okay, so first, we needed to see what are the biological, specifically molecular features that are most associated with doubling time. And we found that gene expression level was by far the most informative feature that we had, much more than mutations or copy numbers or fusion, etc. Gene expression was very informative, and thousands of genes correlated with doubling time, just like the most correlated one. But even after applying many filters, meaning just genes that have a high expression, so we'll be able to measure them, etc., and we still had about like 3,000 genes of their expression correlated with growth rate. So basically, we trained machine learning models with gene expression data. It's going to be the only machine learning figure here. See? No, okay. So basically, we tested different number of features. Now features meaning how many different genes should we apply to the model. And we tested many different models. Very intuitively, no single model outperformed all the other, rather aggregating information from multiple different machine learning and taking their average gave the best results. And here this is like the evaluation on test data from the cell lines. Okay? So at least in cell lines, we are able to infer growth rate, doubling time, in a relatively reasonable accuracy. But before we move to patients, we wanted to see if we can have some kind of a validation outside of the realm of cell lines. And we applied it to PDXs. Again, not exactly what happened in the body, but still already a big leap compared to what we have for cell lines in a petri dish. And we are able to infer it quite well. I didn't mention, I don't have it here. Our always comparison is to the gold standard Ki-67 level. And also in cell lines and in the PDXs, the prediction of DT learner are superior compared to the Ki-67 level. Reasonable because, again, we are measuring the real thing that we are talking about, which is the doubling time and not the proliferation rate. Okay? So then we applied it to patient data, and we wanted to see if we can predict survival. We applied it to the TCGA data set, and we found that in about 30% of the tumor types, we call it PDT, like the predicted doubling time, correlated significantly with survival, which means that it's a useful biomarker. And in some tumors, it was already known that growth rate is correlated with survival, like in lung. But we also find some new ones that was not previously known. More or less is like more expected. We have some other biological features in the patient data that the PDT predictions correlate with. I just want to clarify one thing. We call our measurement PDT, like we give it like as a biomarker, because we don't think that in real patients we actually measured or able to predict the true doubling time, because the situation is very different. Our predictions are basically about 10 times faster than what happened in real patients for growth rate, because in cell lines the growth rate is much faster, but we still treat it as a measurement that is correlated with the true doubling time, okay? So we don't really measure the true doubling time, we measure biomarkers that correlate with the true doubling time. Okay, so this is like what was known. Now I wanted to see if we can predict drug response, because eventually all the other are nice, but what we really want is to be able to stratify patients based on these predictions, just to be able to tell to a patient, you have 5 months on average or 20 months for average. It's not really informative. I know that I don't look very well groomed today. Basically, my mother-in-law passed away last week from cancer, from pancreatic cancer. So she got on average a year, but she survived 4 months. So it doesn't really matter what's the average prediction. We want to be able to do something with the treatment, just that to say what your average prediction. Okay, so we tested on a small data set where we had both gene expression level and drug response. And for about 7 different chemotherapies, we're able to predict response to the treatment. And however, not in treatment that are not expected to be correlated with the growth rate. So basically, we're able to measure it for chemotherapy, but not for targeted and not for immune response. Okay, so we showed that this measurement can be applied to patients. But now you wanted to see on new drugs if we can tell something about them. So basically now we went back to cell lines. We took a data set from DepMap. Not for all of them they had measured growth rate. So for about half of them they didn't. So we used our model to evaluate and predict their growth rate. So now we have like a large data set of about 800 cell lines where we have both growth rate or either measured or predicted, and we have the drug response. And we try to find the correlation of the predicted, so it's not predicted, yeah, of the predicted growth rate and the drug response. And we found quite a bunch, like about 800 of the drugs from the size, I think it was like something like 5,000, that correlated with growth rate. And what I want to actually raise for discussion for the audience, because we don't have any good explanation for that, is not the red ones. The red ones are the drugs that are associated with doubling time in the sense that faster growth rate is associated with stronger response. Because we know many drugs, especially chemotherapies, attack cells while they are growing, not when they're dividing, not when they are senescent. And however, we have a set of drugs, mainly EGFR inhibitors, that are associated more with cell lines that have larger doubling time, meaning they grow slower. So basically, we have a treatment that if you are dividing, it doesn't affect you. However, if you are growing very slowly, it will kill you more. So we don't have any good mechanism to suggest for that observation. We wanted to see if this is like some kind of artifact of our algorithm, so we tested it separately on the cell lines where the doubling time was measured, and separately on the cell line where the doubling time was predicted. And each one of them separately, the EGFR inhibitors were associated more with cell lines with larger doubling time, again, cell lines that grow slower. And we don't have any suggested mechanism, so basically our last, even to suggest a mechanism, our last piece holding us back from submitting this manuscript. Mainly eventually we just give up and submit it without a suggested mechanism, but this is our current situation. So I decided to present this work here today. Maybe someone can give us any suggestion. Anyway, so in terms of all the other drugs, so basically this is like their suggested mechanism. In many of them, it was predicted that the mechanism will be correlated with growth rate. In many of them, not. So, okay, so this was one of the stories that I wanted to predict here, to describe it today, and this is like my lab, and I want to thank the students that were working on the lab, Lin Yu, which was initially working on the lab, and then after he left, Daniel and Geya took over the project. So thank you for listening, and I'm open for questions.
Thank you, Yosi. This was wonderful. Do we have any questions? By the way, you're mistaken evaluating our audience. We have some machine learning scientists here, and the next speaker will be one of them, even though he's a physician. Questions?
Sorry for the audience. Apologize.
Yes.
So super interesting. I have a question about what we can maybe learn from additional information we can learn from the data, because doubling time, you can think of having a longer or shorter doubling time, you can modulate different parts of the cell cycle, right? So do you have a sense if what you're measuring, because Ki67 is a marker for G1. So basically what we're, in the clinic, what we're measuring is how long is your G1, right? So do you have a sense how that?
How many patients are?
Yeah,
how many cells are in G1? Yes.
In G1.
Yeah, but.
Yeah, so no. So maybe you should try to do it. Like basically at the end of the day, our prediction, what is based on. It will be non-trivial because we tested many different combinations of genes, and we found that it had a very minor effect on the prediction. So from this list of 3,000 optional genes, we can pick almost any set of 50 genes from the top 1,000, and the prediction will be very similar. So it can't say, maybe every random set of 50 will pick some genes from many different processes. Maybe we should try to eliminate the genes based on the process, and then it can enable us. But like, I think it's very complex because it's also about death and which step in the cell cycle. So, but okay, it's a good idea. We should try it.
Thank you, Yosi. That was great. So I actually wanted to ask that before, but then you raised a question and it's related. If you look at the features that were important in your machine learning approach to infer doubling time, you can use, for example, Shai value, and you can quantify it. So number one, do you also use Ki67 as a feature? Number two, can you use features that are the lineage, like which cell line it is? And then, for your question, EGFR, you do the same. If you run your machine learning approach to learn what would provide EGFR response or resistance, you can look at overlap, maybe to give some insight into the mechanistic reason it works like that.
Okay, so Ki67 was in the lower part of these 3,000 genes. I don't remember the exact position. I think it was around 2,000, something like that. So it probably didn't get into the models in any of the runs. So maybe we should try to enforce it. But yeah. So, and we also tried to incorporate the information about the lineage for the algorithm that can include this like non-continuous variable. It had barely no effect. The only feature that we found was different was to eliminate or include hematological tumors. So hematological and solid had a difference, but beside that, no. So maybe if instead of 800 cell lines for training, we would have 80,000, probably we would have found some kind of features that are very lineage specific. But in our 800, the only advantage that we found was to try to exclude hematological tumors.
So my question is exactly about that, because in some cases of lymphoma, the patient would not get treated at all until you have a certain burden of the tumor. So maybe this can link you to the actual in vivo in patients data.
Okay. I was not aware of the lymphoma. We tried to test it on CLLs, and we didn't have a large enough cohort. We were able to find a cohort of 20 patients that we had their expression and measured size through time. In this small 20 patients cohort, there was no correlation. But again, CLL is like very slowly growing tumors, and maybe it's like already outside of the range of what we were training on. So we tried to test it. Eventually we stopped because of, like, it wasn't successful. But we don't know if because something is unique about CLL, is it because of just 20 cell lines, 20 patients? So this is the situation.
Out of curiosity, maybe the cohort is too small, but have you ever seen, this is speaking on what Yaara was asking, have you ever seen a contradicting measurements looking at the Ki-67 if it was, let's say, on the lower single-digit percent, and so a contradictive doubling time by the algorithm, because it seems to go together, right?
Okay, so we didn't find a full overlap, but not being T-able significant doesn't mean that it works for the other direction. So there were definitely cases of no overlap. We didn't try to see if there were contradiction between
I'm
them.
wondering if you will, in terms of intervention, will you do something differently if you get the response?
Well, this is like many very like basic proof of concept. It's not something that we are tomorrow morning bringing to the clinic. But
Okay.
yeah.
Fair enough. Thank
Okay,
you.
Thank you.
Now, we're moving to Hadassah now. So our next speaker is Professor Shai Rosenberg towards AI-assisted precision oncology. Thank you. I think it's open, right?
Hi, good morning, and thank you for Ronit for arranging this beautiful conference and inviting me. I will talk about two algorithms that we developed in our lab and that are related to precision oncology. So this is the cancer patients of mine with the glioblastoma. We usually send those patients, now in Hadassah, every patient we send to genomic testing, and we get a profile of mutations. And the state of the art of clinical precision oncology is that looking at this list of mutation and try to match a drug. If we are, of course, outside the standard of care, we try to match a drug, and then we say, okay, let's see if it helps or not. So the patients actually an N equal 1 clinical trial. The situation is less severe, but still in standard of care drug. You can see here the New England Journal of Medicine trial of osimertinib, which is a third generation EGFR inhibitor. And you can see that those patients are responding better, slower progression-free survival. And this is now the drug given to all patients. But you can see that only 80% of the patients respond, and also some of them, eventually all of them, stop to respond to the drug. And if we could have a biomarker to stratify the patients better to those who are more likely to respond as not likely to respond, we would have done better precision oncology. So I want to show one algorithm that tried to address this through a biological mechanism. You probably all know about the mutational signatures. You can see here mutational signatures of tobacco in lung cancer, of UV in melanoma. This is distribution of mutations. But in order to infer this distribution, we usually need all exome sequencing, all genome sequencing, whereas for the patients, we usually do gene panel sequencing, which encompass 1% of the genome. 5, 10 mutations, 15 mutations, it's not enough to infer the signatures. And hence, this is a very fancy science started by Alexandrov and Struton, but it's more biological, and most of the signatures did not reach any clinical utility. Whereas we have a growing number of patients with panels with clinical information, but we cannot apply the signature analysis. So this is the gap that we wanted to address. This work was done by my student, Dr. Adar Yaakov, who is now resident in oncology in Shaarei Tzedek. And before this, I want one small technical note on natural language processing, like ChatGPT and the transformer. They usually try to code the words on a vectorial level. So we can see the dog and cat are close on the y-axis because they are domestic, and wolf and tiger are closer on the y-axis because they are wild type. They are wild animals. But on the x-axis, dog and wolf and tiger and cat are closer because they are the same family, the same taxonomy. So if we could take the language and vectorialize it like in ChatGPT, which is tens of thousands of dimensions, then we can find a very rich contextual relations between those. This is actually how ChatGPT and transformer work, the main part of them. So why tell it to you here? So what we want to do is to code the signatures and code the single mutations on the same vectorial space. So what we did is that we took the TCGA data for which we have the ground truth. We have exome sequencing, we know what the signatures are, and then we had a mutation, and we took not only the 5' and 3', but we took a 5-mer, two nucleotide 5' and 2 nucleotide 3'. So in order to be more specific, it's over 1,500 types of mutations. And then we trained the algorithm using an architecture named star space in order to create a vector, a true vector for the signatures and the mutation that will be closer. So now, how does it look? You can see here, these are 4 mutations. The mutation have vectorial values on 200 dimension. And then when we do the averaging, we can see which signature, which is on the same vectorial space, is closer to the average of the mutation. And therefore, we could train those models in order to identify 9 different signatures with as low as 4 or 6 mutations. What are we doing with it? So now it opened us the way to look at clinical samples. So we took now 1,800 patients from Gini BPC database. It was 2 years ago. And we just analyzed for all the signatures, and we have there all the patient journey, all the treatments, all the response to treatments, the duration of response to treatments, and then we did all the mapping. And we arrived to several conclusions. One of them, interesting, was for EGFR inhibitor. Fortunately, we don't see it here, but we can see that a lung cancer patient receiving EGFR inhibitor with the signature of up or back are not responding to the drug. The putative biological mechanisms that ApoBec enzyme activity causes mutations, more resistance. It's not only for EGFR, but it's more marked for EGFR. But when we wrote the paper, 2 papers in Nature and Nature Genetics appeared that identified the same signal but from a completely independent point of view. They did an experimental work, and this was for us a very good validation, independent validation. Of course, we have a lot of other signals that we are now validating with additional sets of patients that we are now writing a second manuscript on. The second algorithm, so this was the first algorithm, to summarize, the first algorithm that identified the signature which was not possible to do before, and then it opened us the way to test the clinical utility of the signatures and to identify patients more likely or less likely to respond. Now I want to move to the second algorithm. And the first, I will describe the basic computational biology out of it. It was a work done by my student, Dr. Kobi Lando, who is now a resident in nephrology. He decided to went to nephrology, but he's a cancer researcher. It was published 2 years ago. And then we ask about dichotomy. Usually we treat mutation in dichotomic way. We say either there is a mutation or there is no mutation, TP53 mutant or not. But we see, if we look at the landscape of TCGA patient, we see that there are some mutations that are much more prevalent. And we ask ourselves why. And there can be 2 reasons. We hypothesize it can be 2 reasons. First of all, maybe the mutation rate or stability in those regions are higher, and there are just more mutation. The mechanism of the mutagenesis is higher, or those mutations have a selective advantage, a better selective advantage compared to the other mutations. And what we did, and we did a calculation that we had the mutational rate for every possible position in the cancer context. And then we use it to normalize the mutation prevalence of in large cohort of over 140,000 patients. And we arrived to a statistic named tumor variant amplitude. This is a logarithmic scales. And 2 means that the mutation is 100 more prevalent than expected by neutral evolution. There are a lot of parallelism between classical population genetics and cancer genomics. It's a little underrated, but there is. And we compared our TEVA, for example, for TP53, we compared it to a score published in Nature Genetics 2018, to a functional score of mutant transfected to cell lines, when the score is higher than the disruption of the gene, of what protein is high, and we arrived to a very high correlation between our score, experimental score, and their score. We also saw that there are weak, medium, and strong level mutation, which is not dichotomic. But now our novelty is that we can do it computationally for every cancer gene that we want. We don't need a $2 million experiment for it. So now we took TP53, for example. We want to validate it in different biological systems. So first of all, survival. We divided all the TCGA data into patients stratified according to the TP53 mutation, and the patient with the stronger mutation died faster than the patient with the medium mutation, and the patient with the weak mutation acted actually as if they don't have a mutation at all. So we're very excited to see that our simple computational biology addresses or is informative in terms of the ultimate and most complex phenotype of survival. For some reason, one of the slides is we showed it also in POL-E gene that cause a lot of mutations, and the stronger mutation in POL-E, which is related to DNA repair, the stronger mutation in the POL-E causes much more mutation in the tumor. So it's the third system. And we also show that this is, for example, look only on the upper left panel. This is from Cell Line Encyclopedia, a PIK3CA are stratified here according to the strength of the mutation. And this is the IC50 for the inhibitor. And we can say this is logarithmic, so it's much steeper than it looks, that stronger mutations are associated with better response. Now, this was very strange for us because this measure is not trained on pharmacological data. It's a biological measure. And our hypothesis, which we didn't prove yet, but this is our hypothesis, we do research in order to validate it, is if there is a stronger mutation than the tumor, and this is what we saw, the tumor tends to have less other mutation because it doesn't need it needs less alternative mechanism. So if there is a stronger mutation, it might be that this mutation is the Achilles heel of the tumor. So if we target the strongest mutation, then we do a lot of we do much more benefit for the patient because this is the Achilles heel. This is an hypothesis. How we can use it? We can use it here. We can see a list of mutations. We can stratify the mutation according to the strong, the medium, and the weak mutation. Then try to prescribe a drug according to the priorities given by such computational biology measure. This is also needs to be proven. It's an hypothesis here. The clinical application is still an hypothesis, but we are doing work with it. But the problem, what was our bottleneck, is that we were limited by the information that was available for us on the mutation, because we had 140,000 mutation, and patients, and only most prevalent mutation we get a reliable measure of this tumor variant amplitude. But most of the mutations are rare, and we didn't have conclusive information about them. And here comes the new work that is unpublished yet, that we do with Professor Ora Furman from our Faculty of Medicine, a computational structural biologist, and my PhD students, Noah Klugman, who is part of the Sagol program for computational medicine. My students are doing a dual degree in medicine and computer science. And we took a different question, because usually we now have AlphaFold and AlphaMissense. AlphaMissense and Eve and other leading algorithms can classify passengers and drivers. This is a very saturated field of research and is not very, very interesting. They do it quite well. But no one is stratifying the mutation according to weak driver and strong driver. So according to AlphaMissense, if it's a driver, it's a driver. It's a dichotomic. This is how AlphaMissense was trained. We're trying to see if we can train to discern between weak driver and strong driving. So we took these 11,000 mutations that we have enough computational biology and evolutionary data, and we created, and this was the synergism that I asked Ora to collaborate with me. We created 18 structural parameters, because our computational biology thinking was evolutionary. We saw what the tumor like, which mutation the tumor like, and we do the calculation, validation. But if we take a completely orthogonal field of information like structural biology, it might be very interesting. So we created 18 structural parameters. This is one structural parameter, the only structural parameter that I invented, because the others are Ora invented, they are existing. We created a sphere around every nucleotide, and then we measured the TVA, the tumor variant amplitude value, for the mutation that we know. And then when we average a code around this sphere, we have a computational biology measure, a structural measure about the TVA, even though we don't have a TVA for this position, this nucleotide, but we know what is the relative importance in the area of this algorithm. And then we trained classical machine learning algorithms, and we arrived in the test set, we arrived at 0.89 area under the curve to discern between weak and strong driver. We did the feature importance and the maximal TVA in position, the weighted sum of TVA in this sphere are the most informative one, but they are not enough. We need the other parameters. The distribution of strong and weak mutation is this on the other on cancer share datasets from a lot of sources in TCGA. We have shown that the strong mutations are more clonal. So it's also in accordance with our hypothesis that they have a stronger selective advantage. We have shown it also in we repeated the same PIK3CA experiment, and it also worked there. Just in the last minute, I want to show what are we doing it clinically in Hadassah. So in Hadassah, we have a project with Roche. We've sequenced all patients in Hadassah, all cancer patients in Hadassah. We created the data structure of patient journey that is automatically curated. I did it with our IT team, automatically curated from the databases of Hadassah. You can see here all the treatments and radiation, diagnosis, surgeries, and clinical notes in the same screen. All the doctors are now doing it. And we created, with the Israel Innovative Authority, we created a data mart of 260,000 cancer diagnoses since the end of the 70s in Hadassah that is based on this mapping, and it's updated every week for all living patients automatically. So we can retrieve it, we can play with it, and now we have sequencing for all new patients, now currently more than 300. So we have this infrastructure environment in Hadassah of doing the genomics. We have the clinical data, and then we can develop AI algorithm, but actually test them and stream the data for the patients. And with this, I will conclude. And this is my thanks to all my collaborators and funders, and I'll be happy to take some questions.
Thank you. Thank you, Shai. I found it fascinating, especially that I was a bit puzzled when you called the BRCA1 mutant as a weak mutation. Recently we were looking at p53 inactivation by chromosome, the short-term loss of chromosome 17p, where p53 resides in breast cancer in primary, and we saw that those that have the inactivation have brain tropic to go to the brain, and then to create metastasis that the patient then succumb to the disease. So I wonder if you were looking only in glioblastoma or also brain metastasis or agnostic to any tumor when we.
So I have two things to say. First of all, I'm treating neuro-oncologists, but as cancer researchers, I'm doing pan-cancer work because we need a lot of data. So I don't want to focus only on specific cancer type. So it's a pan-cancer work. Regarding p53, I think I will be very happy to try to see if we can find a mutual collaboration on this, because the p53, the findings that you find, and to stratify them according to the strength of the mutation, which is usually not dichotomic. I have one thing to say about BRCA. BRCA, interestingly, is usually not a somatic mutation in breast cancer. It's most germline. I asked myself why. And one of the hypotheses that I have, and this is a manuscript that we write now about different germline genetics, is that BRCA does not need to be a strong mutation because it exists in all the bodies of the cells of the body. So it just sits there and waits for 70, for 80 years for an opportunity to become cancer. But somatic mutation is happening in only one cell that is under a lot of microenvironment and immune stress, and it has to be stronger. So this is an hypothesis. I didn't prove it yet, but I think that mutations that are only germline can let themselves be weaker. So this is some hypothesis that
weaker, but very aggressive when they wake up, when they are no longer persistent or dormant. Yeah. Any questions?
Okay, thank you.
So thank you. So speaking on BRCA and taking it to breast cancer, I'm really happy to invite Professor Einav Nili Gal-Yam. Sorry. So there is a mistake here. From Sheba Medical Center, we'll speak about microbiome dynamics in breast cancer, from tumor ecology to dietary intervention. Thank you, Einav.
Okay, still good morning or good afternoon. I need to get out here. כן, אני מחפשת את המצגת שלי. אוקיי. Good. Okay. So thank you for inviting me to join, and I will take you to another direction. So we'll go on a clinical path and not a molecular path. So we'll talk about microbiome dynamics in breast cancer patients. And as you probably all know, well, I'll do it like this. Yeah. So breast cancer is one of the most common cancers, and it affects 1 out of 8 women. 2.3 million new breast cancer patients will be diagnosed, and 100,000 deaths from breast cancer each year, and 25% of new cancer cases are in females. So in terms of risk factors for breast cancer, so aside from the genetic and the family history, which are the main risk factors, and age. As we age, we are prone to get breast cancer. So the environmental factors are very important, mostly sedentary lifestyle, obesity, we'll talk about that later, alcohol consumption, and smoking to a low extent, but they are significant. Of course, parity, childbearing, and exposure to estrogens. All these lifestyle and environmental factors may have a very strong correlation or interplay with our microbiome, a very large organ in our body. So let's talk about gut microbiome and cancer. So our gut microbiome is a very important organ in terms of immune modulation, metabolic changes, and inflammation throughout our lifetime. And it has been found to correlate with cancer progression in terms of dysbiosis. So dysbiosis, changes in the components of the gut microbiomes, were found to be correlated with carcinogenesis, tumor progression, and therapy resistance. So what do we know about gut microbiome and breast cancer currently? We know that microbiota of women with breast cancers differs from that of healthy women, but we have very small cohorts until now. We see dysbiosis, which is an imbalance in the gut microbiome, which is associated with breast cancer risk. And importantly, we know that gut microbiota can modulate estrogen serum levels by bacterial genes that produce estrogen metabolizing enzyme by beta-glucuronidation. So estrogen goes back to the circulation, and estrogen levels are higher. However, most of the studies involve small cohorts and limited clinical information, which is crucial to understand the multifactorial connections. And so we embarked on our Sheba local gut and tumor microbiome project. And we asked, do we know or can we see a role for microbiome in breast cancer? So we do it in two directions. One, with a collaboration with Professor Eran Segal from the Weizmann Institute, we research the gut microbiome in breast cancer, and also tumor microbiome with collaboration with Professor Ravit Straussman, but I will not talk about that today. And the two people which are mainly, or did all the work, are Jonathan Studix, which is an MD, very talented MD student from the Technion in the ERROR project in Sheba Medical Center, and Dr. Maya Dadiani, who actually heads the lab. So the breast cancer gut microbiome study in the Sheba. So we collected around 240 patients and performed longitudinal multi-omic analysis on these patients, collecting stools, urine, blood, body measurements, anthropometrics, and clinical data and questionnaires. We recruited 100 patients, which we were just diagnosed just after the operation, after the surgery. So these are all early disease patients, and 140 patients which received treatment or which had undergone intervention, dietary intervention. And we looked at two things. First of all, we wanted to compare our patients, our breast cancer patients, so this is a very large cohort compared to what was described in the literature until now, to a larger cohort of healthy women. For this, we took advantage of the Human Phenotype Breast project, previously also known as the 10K project, which is running in the Weizmann Institute and is a very, very, very large, and now it's multinational, a large-scale deep phenotype prospective profiling project which collects multi-omics and microbiome data and genetics and immune profiling along the years, okay? So they've collected already about, I think, 20,000 or 13,000 patients and many, many, many samples, and this is ongoing in various countries already. And we wanted to compare our cohort to this, and we'll talk about that. Also, we wanted to look at longitudinal analysis. What happens to the microbiome when our patients get therapy, either chemotherapy, that's a small amount of patients, but mostly endocrine therapy. These patients have metabolic changes. We want to see how this correlates with the microbiome. Also, we performed a clinical trial, which I'll tell you about, which we performed dietary intervention in our endocrine-treated patients. So these are the cohorts. So we took a 1 to 10 ratio from our patients to healthy women. So this will make, or will lower the noise and will make our results much more significant. So there's about 100, half, 100 of patients just newly diagnosed and 100 patients that were already receiving treatment, but these are breast cancer, early breast cancer patients with no evidence of disease, and 2,000 women, which were matched for age and BMI. Why? Because we know that age is a contributor to the changes in the microbiome. We wanted to normalize for that, and also, obviously, BMI, obesity, and would normalize for metabolic changes. Interestingly, when we look at the in clinical parameters of these two cohorts, at the baseline, we see that they are quite similar in terms of triglycerides, LDL, HDL, hemoglobin A1C, even weight. When we look at the clinical parameters of parity and breastfeeding, we see that these are significantly lower in a breast cancer cohort, as expected, but it is nice to see that this really reflects a truly bona fide higher risk cohort. So what about the microbiome? We see that our breast cancer patients have distinct microbiome features compared to the healthy controls. First of all, they have lower diversity, so there's this measure of diversity. Okay, alpha diversity, richness of the microbiome of the species and the phylums and the genuses. It should be diverse. When the diversity goes down, this is usually correlated with pathological conditions, same as Shannon, the index on the middle, so it's evenness and richness, and also the F2B, the Firmicutes to Bacteroidetes ratio, which is lower in pathological conditions. You see that in the breast cancer patients, these indices are lower than compared to the healthy controls. So these are key features of dysbiosis. We see dysbiosis in our patients. What about when we delve deeper in the phylum level? So in red and orange, you see these B to F ratio, Bacteroidetes and Firmicutes, and you see the change in the healthy compared to the breast cancer patients. But if you look at the little green line just in the bottom, this is actinobacteria. Actually, this is one that changes twofold. It's difficult to see, but then it's blown out there. So we see enrichment in this phylum in our breast cancer patients. When we look even deeper, we see that in terms of genus level, so Bifidobacterium, they are enriched in our breast cancer patients. These are our main genuses in this actinobacteria phylum. So we were asking, and the guys at Weizmann, together with Jonathan, were asking, can we actually create a signature or divide, create a signature for patients, a microbiome signature, which will define, which will recognize breast cancer patients as compared to healthy controls. And the AI-based machine learning prediction model was used, and as you can see, so all the microbiome results, so non-supervised, were thrown into the model, and the model successfully, with an AUC of 0.8, could divide to actually predict breast cancer patients compared to healthy controls. So this is just a cross-training model, so this was not validated on an independent set, but this is considered an excellent prediction. And the question is, can be used later or in the future to screen patients at risk. So when we look what contributed to this model, okay, what are the main species that contributed to this model? Again, we see these bifidobacteria species. And why is it interesting? You see these in pink on the left side, and on the right side in the box plots, we see enrichment of bifidobacteria in breast cancer patients. Why is it interesting? Because actually bifidobacterium, you all know these bacteria, right? They are actually considered quite beneficial. They're found in yogurts. They are supposed to be protective. They are metabolized or create short fatty chain acids, and actually their role in cancer as a whole is supposed to be protective, anti-inflammatory, and it was quite surprising to see this enrichment in the breast cancer patients. When we looked at clinical data, so this is just one plot, and we looked at our patients because we have follow-up for about now 5 years, median follow-up. So these patients are really good. So we've got very low number of events, but still when we divide the patients to high versus low bifidobacterium enriched, then we see that the survival, or the DFS, disease-free survival of the patients that have a high bifidobacterium is lower, and is significantly lower than that of those with the low. And this is also after doing multivariate analysis for T-stage and lymph node status, as you can see on the right. What happens after chemotherapy? Bifidobacterium was significantly reduced, and this was adjusted for genus, age, and hormonal status. What happened after endocrine therapy? So these patients get endocrine therapy for months and even years. So we know that on a clinical level, we see decreases in LDL, which is beneficial for the patients, but the bifidobacterium are anti-correlated to this decrease. When we look at other species during endocrine treatment, so actually quite funny, we didn't see many species that were actually changing. So our microbiome is pretty stable over the time of endocrine treatment, but we did see one species was increased, or was significantly increased. This is a Blauschia species, and also this was adjusted for age, chemotherapy, type of treatment of the endocrine treatment, and we saw this increase, and we know that also Blauschia is involved in short-chain fatty acids metabolism, regulating immune responses, and promotes anti-tumor immune responses in various cancer models, so we need to see what effect it will have here. So summary one, gut microbiome of breast cancer patients have lower diversity and lower richness, indicative of dysbiosis. We can provide a microbial signature that can distinguish breast cancer from healthy women. May this be a reflection of the risk to develop breast cancer? Is this just correlated? This needs to be validated, obviously, in prospective cohorts and used, and maybe or possibly used in future, if validated, to screen patients at risk. We do see high abundance of bifidobacteria associated with breast cancer compared to healthy women, related to worse survival. Is this a conundrum? We still need to work this out. And identified species associated with endocrine treatment. And well, I don't see the time. So 5 minutes, good. So I will switch gears here to talk about breast cancer and obesity and our diet intervention trials. So breast cancer, apart from being a genetic and family history and so forth, is an obesity-related cancer. So it's one of the 13 cancers which are related to obesity. So obese patients, obese women mostly, have a higher propensity to develop breast cancer. And if they are obese, their prognosis is worse. Their disease-free survival is shorter, overall survival is shorter. And so diet and nutrition represent modifiable factors that may influence the recurrence, the comorbidities, and the quality of life, because these patients actually complain a lot, first of all, that they gain, a lot of them are obese to start with, but that they also gain weight on the endocrine treatment, which actually hampers their adherence to the treatment. So we said, okay, we'll take all that and do an intervention trial. So this is called the Breast Cancer Personalized Nutrition Study, the Breakpoint trial. This is the phase 2 trial. What we did, we took our patients that they were on endocrine treatment and complained about weight gain. So these were not patients who were happily thin. And we divided them into 2 groups with a diet intervention. So it's not a no intervention versus intervention. Both groups got diet intervention, nutrition intervention. One of them was just the Mediterranean diet, as we all know it. And one of the other arm of the intervention was what we called the postprandial, the personalized postprandial targeting, glucose targeting diet. This was developed also in the Weizmann Institute, and Dr. Michal Ryan, was a PhD student at the time and a dietitian. She followed and treated actually the patients in Sheba Medical Center. And this diet, the PPT diet, as we called it, is based actually on microbiome profiling of the patients and profiling their glucose response to various foods. And the idea, the theory is that based on the microbiome composition and other factors, the response of specific people to various foods is different, and this is reflected by the glucose postprandial. And so let's target this so that the glucose levels will not be as high. And so they got a specific, for each one, a specific menu, which was designed by Michal, and the control group, the Mediterranean diet. And what we wanted to see, first of all, will we see loss, weight loss? Will it be different between the 2 arms? Will the glucose control be different between 2 arms? And let's look and see what happens with the microbiome. So in terms of our cohort, these were mostly, so it was 127 patients, 60% were postmenopausal, and about 60% were overweight, over 27 for BMI, and we controlled for chemotherapy and hormonal therapy and tumor stage. So these was stratified. So short look at the results. You see all these patients prior to, so this is the gray bar days prior to starting the trial, they all gain weight, and then when they started at average similar weight, both arms, and they all lost weight, which was good. Patients were very happy, coming to me and saying, wow, so nice, they got a free dietitian for 6 months, every month, and they lost weight. They had this logbook, they entered all what they ate. It wasn't a very significant, it wasn't like GLP-1 analogs today, it was about 3-4% weight change. And it was similar between 2 groups. So Mediterranean arm and the personalized PPT did exactly the same in 6 months. In terms of waist circumference, which is very important in terms of fat, in the visceral fat, there was a better response, or seemed to be a better response, although not statistically significant for the PPT arm. What's more important, though, is when we stopped the intervention, and then people continued their diet on their own for another 6 months. And we know for everybody who's been dieting that this is the hard part, right? After 6 months, now we need to maintain weight. So the PPT arm was able to maintain their weight and the waist circumference much better than the Mediterranean arm. So, of course, everything is funding. So in terms of weight, we stopped looking at 12 months. So we don't have, but some of them keep coming to me, and some of them actually do maintain their weight and are very happy about it. So when we look at other factors, in terms of metabolic age, visceral level, and fat percent, all of these dropped in the 2 arms, but this was more significantly so in the PPT arm. What about changes in the glucose, in triglycerides, in cholesterol? So the PPT diet does what it is supposed to do. So it lowers glucose after meals. So you can see that all the red, okay, the red lines there in the B, in paragraph B, and also the PPGR. So these are relevant to glucose measures. They were much better. Postprandial glucose levels were much better, much lower in the PPT arm, and this may be related to the propensity to develop diabetes later. And this, we know, diabetes, metabolic syndrome, these are, first of all, we see it in breast cancer patients, and these are prognostic or bad prognostic factors in our patients. So in terms of other factors, cholesterol, triglycerides, and so on and so forth, there was no change. So they were lowered, but there was no change between the 2, no difference, sorry, between the 2 arms. So, last thing, what are the factors related to a reduction? So if they received chemotherapy or menopausal status or age or even the study arm was not statistically significant, but if they received tamoxifen, they had a much more difficult time to lose weight compared to aromatase inhibitors. And we actually see it in the clinic, but this is something that we now saw in the numbers. Microbiome, I really hoped to give you also the microbiome changes and then close the loop with the Blauschia and the bifidobacteria story. Unfortunately, these are still in analysis, so next year, BRC meeting, I'll report that. So to summarize here, we see significant weight loss in both arms with no significant difference between the arms. Both dietary interventions improve anthropometric outcomes. The PPT diet improved glucose parameters, and at 12 months was associated with improved metabolic-related anthropometrics. Type of endocrine treatment was significantly correlated with weight loss, and the microbiome dynamics post and pre are analyzed. We wanted to see the interplay between the microbiomes and the type of diet and the weight loss. So now I will say thank you for staying this late, and thank you for all the teams that took part in these studies. Thanks.
Thank you. Do you have an idea if anyone was taking GLP-1 or any
Not yet.
correlation to?
Not yet, not yet.
No.
So this is before the era. So GLP-1s, GLP-1 analogs, really entered like 2 years ago, I think, and now a lot of our patients actually wanted to take it, and there's no problem with that. And what has been seen so far, there's no increased risk when taking GLP-1 analogs, and actually. This was, we wanted to see what will happen with these patients, with the GLP-1 analog patients regarding the microbiome, endocrine, adherence, and prognosis. Yeah.
Interesting.
Yeah.
Questions? Yes.
Ofir.
That was fantastic. Thank you. So I'm wondering if you think about the microbiome signature to predict breast cancer and to know if it's like causative and can predict. Is it there, again, far from my field, but you probably know, is there a biobank of stored stools or samples? And then maybe that could be assessed to use the time to event models, correcting for age, BMI, and so on, and see if that kind of classifier can give you a handle.
Yeah, obviously, this is a great question. So we are not aware of that. So that's why we are collecting for breast cancer patients. But the HPP project, as I told you, you're asking about the patients, or you're asking about healthy people?
No, healthy people. Like
exactly.
If there is a biobank of stored stools, then that question can be.
Okay, so this, yeah, Marim Lanchata. HPP, so the HPP, the 10K project, okay, so it is following these people, these healthy people, which will probably, some of them, develop cancer sometime. And the idea is that using the signature that now was developed is, and to see if patients that eventually develop cancer, and there are a lot of, these are a lot of people, will show, or maybe we can refine even the signature. So this is being done, following, yeah.
Yeah, I was wondering with the cachexia.
Actually, I was going in that direction, but you're right. Do you know, you have any idea which metabolites are eventually associated with this?
Yeah, so another excellent, so we're doing metabolomics. Okay, so these are currently being performed, these analyses. So both, yeah, so two kind of metabolomics. These are performed now in the Weizmann, and so we should get the data and analyze it. So we don't have the data yet. We have it for the tumor microbiome, but this is a completely different story. Yeah, but it's complementary. It's an excellent question.
A bit of a trivial question. Would you recommend now a bifidobacterium-rich diet for the general population or cancer, breast
so
cancer
it's like I was talking with Maya. I said bifidobacterium. So I was trying. I was thinking that I need to say, don't stop buying your yogurt just now, okay? Because, I mean, yeah, we don't know. We actually. So this is a very surprising. So when you see such a surprising actually result, first thing you think is a trill, right? So we'll go back and see. But there are very, a lot of points of data that combine together, and it looks very real, but now we need to see what it means.
And going back a bit to BRCA that we mentioned before, do you see any difference between BRCA carriers?
So that's another excellent question. So this cohort was not enriched for BRCA. Most BRCA are 1, and these are postmenopausal endocrine, but we are doing a similar project or contemplating at least doing a similar project specifically on BRCA carriers and to see if the signatures there would, yeah, exactly. So this is a modifier for the risk in BRCA carriers. Not all of them actually develop cancer, right? Yeah. So yeah, but these are very, very expensive, trials. So.
So now we have grants for that.
Right.
Okay, so thank you.
Thank you, and thank you for staying up.
And the wonderful Muhammad Jbara will not be here, unfortunately, today. We wish him good health. And I would like to adjourn with thanking everyone in the audience, all the speakers, the sponsors, MERCK and TEVA, thank you so much, the organizing committee, and the students who took part in this and will continue to work on all this one fantastic project. Judith, thank you to both. See you next year.